I'll mute myself.
Yeah, we are live now, Professor.
Perfect. Okay, great. Thank you very much. Thank you very much, everybody. I see that the attendee list is populating. So this is the short course on Bayesian modeling of brain imaging data. And I'm really glad to be here for, the short courses for ISBA 2021. The short courses are a staple of the ISBA World Meeting. And I'm looking forward to the ISBA World Meeting starting next week. I would like to thank Mingui Chen and Lima and all the organizing committee for organizing this conference next week. It's fantastic. It's a free conference, 2,100 and plus, and increasing attendance. It's just something extraordinary that they've been able to do. So I'm going to share my screen now. And first, I will start by telling you that, so first, you can obtain the slides for this short course online. Let me see if I can, 1 second, make sure that I can see the chat. So by just going to these two links here, you should be able to download the slides for the webinar. So I will leave them here for a second while I start. I want to organize my, 1 second, Zoom meeting. I don't see. The controls. Okay, good, got it. 1 second. Let's try to stop the sharing the screen. Try again. Okay, much better. Okay, so in terms of organization of the webinar today, I will most likely have sessions of 50 minutes and then probably 10 minutes break every hour. We are here together for 10 hours, so I will try to pace the webinar this way. If you have at any time questions, you are free to add your questions in the Q&A section of the of Zoom. I will review the questions from time to time, and if possible, I will also try sometimes, if you are amenable, if you want to have you maybe also ask the questions directly if you are okay with that. Okay, so one starting point is that this is I mean, the past few years have been funded by a National Science Foundation grant. So I would like to acknowledge the support of NSF for delving into the study of brain imaging data. So again, I will put these again from time to time, just in case you want to follow the slides with me. So there is definitely a large interest today in the brain imaging, and this is probably the reason why, oops, you are here today. So the as you can see, it's all over the news. This is just a representative of different types of articles in the news, in journals about the something related studies, reporting studies related to the brain. For example, it's very well known and it made a lot of news, how football or some sports affect our brains. So in this webinar, in this short course, we'll assume the knowledge of some Bayesian tools. I will not assume knowledge of techniques related to imaging, and I will try to go a bit over a few particular characteristics of especially two types of techniques of brain imaging. So there is, of course, statistical methods play a crucial role in the analysis of brain imaging data. However, I have to say that there are not many statisticians that actually work with brain imaging data. At the same time, the imaging community, neuroimaging community, the neuroscientists are very much interested in interfacing with statisticians. So one of the reasons why I am doing this short course is also to have a possibility to provide you with what I think is the status of the statistical thoughts in brain imaging analysis, so that maybe you are going to be involved or going to think about being involved into the field. An important point that I want to make from the beginning is that statistical methods may depend and depend often specifically on the type of technology that it is employed to obtain the data. So in particular, we can distinguish the brain imaging technologies into two major categories. One is what we will call structural brain imaging, and another is what we will actually look more in depth into, which goes under the name of functional brain imaging. So each category spans a number of specific technologies, and each technology somehow commands a specific tool for statistical tool or set of statistical tools to use for the analysis. So in particular, when we talk about structural brain imaging, we are talking about images that are the study of an anatomical structure of the brain. For example, in order to assist the diagnosis of brain injuries or the diagnosis of certain diseases. Think about the example of football before. And typically we have here a few types of technologies that are used the most. One, for example, is computed axial tomography, CT scans, that provide images of bones, soft tissues, and hair. We have MRI scan, where an individual is put inside a scanner, and we obtain basically detailed images of organs. And then we have positron emission tomography, PET, images, where basically we obtain images after injection of a radio tracer. So just to give an idea of the type of field where these, you can find these type of images, for example, CT and MRI scan are very much used in the emerging topic of radiomics, where one is actually interested in studying for example, the characteristics of of bones or soft tissues. It's often also related with cancer emergence, cancer disease. PET is actually often used in Alzheimer's disease studies, for example. So we will not discuss techniques for structural brain imaging studies. We will discuss techniques most often for functional brain imaging. This is actually what I have dedicated part of my research upon in the last few years. And here we can distinguish a few techniques of reference. In particular, the idea behind this type of the type of studies employing these techniques is that we are interested in study brain functioning in terms both of a specialization of the brain, which parts of the brain responds to a particular cognitive or sensory challenge, and also in terms of integration, that is how different brain areas, brain regions interact with each other. So the typical techniques that are considered in this case are EEG. EEG are a very, if you want, simple, not invasive, not very costly technique to record electrical activity in the brain. Basically, the idea is that electrodes are placed along the scalp, and they typically are used for diagnostic purposes, to identify, in the study of epilepsy, in the study of sleep disorders, and so on. It is also used to identify, like in fMRI studies, regions of the brain that maybe, or behaviors, or how the brain reacts to something, for example, shown on a screen, or some stimuli, external stimuli in general. The functional MRI technique is also a non-invasive technique, and is also used to map brain activity, and in particular to show which parts of the brain are involved in a particular mental process. It is a bit more costly in terms of experiments, because the experiments are conducted in a MRI scanner. So an individual is put in a scanner, and then their activity is recorded. There are other techniques, for example, magnetoencephalography technique. So it is also a non-invasive technique that measures the magnetic field around the head to reconstruct the electrical neural activity in the brain. So we have, in particular, all these technologies are different in some specific characteristics. For example, we will discuss in a second, EEG are have high resolution, high temporal resolution, but they have low spatial resolution. On the other hand, functional MRI methods, techniques, they lead to data with a high spatial resolution, but with a low temporal resolution, due to the way in which the data are taken. So let me check one sec if there are questions. And again, I'm having problem with, okay, here, for some reason. I didn't see questions. For some reason, these, okay. For some reason, I lose track of the Zoom controls, so I apologize for that. So before we start and go into the details of the webinar, right? Let me give you an outline of this course, so that also, if you are interested in some parts, you may try to connect at some point during the course. So most of the course will be spent on the statistical analysis of fMRI data in particular. And this is due to the fact that this is an active area of research, but also it is something that I have been working on more closely in the past few years. In particular, I will spend a good chunk of the first hour on basically describing the data, the challenges, and also the type of models that are typically employed. This will be also typically at the beginning, we'll discuss a bit of the frequentist models, because although they are frequentist, right, they are actually adopted, and they form the base of also Bayesian developments, because they respond to discussions that have been conducted in the field. So After that, I will spend some time on Bayesian modeling of brain activations for single subject fMRI data. So the analysis of data from a single subject, and I will discuss a few points that come up, a few challenges that come up also in that context. I will discuss the multi-subject modeling and brain connectivity following up. This will probably be later today. I expect toward probably 3 and 4, probably or the third and fourth hour. And then I will discuss, if I have time, statistical analysis of EEG data and the contributions in the area. And finally, if I have time, and probably I might not have time, I would like to discuss a bit of points or questions related to multimodal imaging, in particular, some examples in the context of imaging genetics. So, let's start with the statistical analysis of fMRI data. And in particular, let's start from a look at the data. We don't have time to go into the details of the of how these experiments work, but basically what happens is that an individual is called into an experiment, and the individual is put into a scanner, and during the experiment, they are asked to perform some simple task, or often, or just lay in the scanner. And basically, in the meantime, their activity is recorded over time. Typically, what happens is that you have a scanner, right, that goes up and down the head, and every 2 seconds, the scanner obtains an entire picture of the brain, as measured by the levels of blood oxygenation level response, what is called the blood oxygenation level dependent response, BOLD response, which is captured by the scanner. So a first point that I want to make is that the fMRI activity, the fMRI data do not capture activity at the neuronal level. What the fMRI data capture is indeed the brain activity as changes in the blood flow. And typically, these changes are captured during a sensory motor task. For example, the individual is in the scanner and asked to see different types of pictures. Pictures, for example, of prairies, and then followed by pictures of beaches, right? And or pictures of food. And basically, the activity of the brain in response to the pictures being shown is going to be recorded. There are also more complex tasks in which the individual is asked to press a button or do simple tasks while for example, seeing pictures or listening to a blip from time to time, a particular tune. From a statistical perspective, what we have is that every 2 seconds, we get an entire image of the brain. And so for each point in the brain, a point in the brain, we will call it from now voxel, a location in the brain. From each location in the brain, we're going to have a time series that represents the activity of the individual in the scanner. So at a basic level, what we have with fMRI data is the analysis of big data characterized by high spatial and spatial resolution, and also observed over time. So we have indeed a sequence of 3D MR images. Every 2 seconds, we basically get an image of 100,000 voxels, let's say, each. And these are typically collected in a single experiment for a single subject, and we get basically 1,000, between 100 and 1,000 images over time, over a typical experiment. So we have a big data problem, indeed, due to the sheer amount of data that we have. Also, a study may involve multiple subjects. Typically, the number of subjects is between 10 and 50 in a typical fMRI study. Remember, the fMRI study is costly to perform due to the use of scanners. An important point is that the signal is relatively weak because it's the signal capturing the blood flow oxygenation, right? Due to the scanner. So overall, the data exhibit a complicated spatio-temporal structure, which we want to use to try to predict, to understand the brain activity, but also eventually to predict and help in the assessment of psychological or clinical states. Let's see. So, as I mentioned, the fMRI data are spatio-temporal objects, right? So, in particular, the temporal resolution of fMRI data is typically low. We record an entire volume, right, every 2 seconds, right? The temporal resolution allows us to separate brain events over time. So it is basically determined by how quickly we acquire each image through the scanner. And typically one important feature, characteristic to take into account is what is called the TR, that is the repetition time between volumes, that typically is between 2 and 3 seconds. So we get an entire image of the brain every 2 seconds. The spatial resolution allows us to distinguish changes in brain activity across different spatial locations. For example, in response to a task. For example, here we can see two examples from a published paper where basically we can identify different regions of the brain where some regions are activated in respect to resting state where the individual is in the scanner without doing anything. So in this regard, as I mentioned, the fMRI data have high spatial resolution, so we can use them to go into the details of the spatial organization of the brain. They have higher spatial resolution than, for example, PET, EEG, and MEG data, but less than structural MRI. And as I mentioned, fMRI data have high spatial resolution, but overall lower temporal resolution with respect to other other techniques that allow obtaining images of the brain over time. And in particular, the spatial resolution, one important point that we have to keep in mind is that when we get typically this data as statisticians, we get this data as at the end of a long process of pre-processing of the data. And this pre-processing of the data involves a lot of steps that may affect eventually the spatial resolution of the images. And in particular, there is typically a pipeline that affects the data that eventually we get. So a few factors that we need to consider, right, in the analysis of fMRI data. The data are highly dimensional. The dimensionality of the data is very large. And of course, we typically have also multi-subject experiments. Of course, an individual is not fixed and is not stable, completely firm in a scanner for the 5 minutes of an experiment. So typically, there may be some acquisition artifacts due to the fact that people move in the scanner, and also to the fact that we are humans. So there are physiological processes that happen naturally, like, for example, breathing, that, of course, are going to affect the our position, right? And also our position in the scanner. We're not going to be completely still, but also the type of things that the scanner is going to see. And then there are some artifacts that are due to the scanner. For example, there is some slow drift of signal across time. There is some instability and so on. All these things needs to be corrected in the pre-processing steps that I was describing above in order to give us a sort of cleaned data. And from a statistical perspective, it is also important to recognize that there is a high heterogeneity between individuals, and there is a lot of variability across time also within individuals. This is something that we will discuss going forward. But before I get to that, I just wanted to make a further point on the type of fMRI pre-processing, so that the data have before getting to the statistical analyst. And in particular, there is basically a pipeline of steps that are typically taken in order to adapt or normalize, let's say, each brain data for each individual in this over this course of experiment, and also to try to adapt and set up basically different types of, so basically to make sure that an individual brain can be compared with the brain of another individual, which is not an easy task itself. So I'm not going to go into the details of these steps, but for example, these steps involve the basically going from the data that are obtained from the scanner, basically to the basically data that we can use for the image through, for example, Fourier transform. There is a nice chapter by Daniel Rowe in the Handbook of Neuroimaging Data Analysis that describes all these steps, and in particular these steps of image reconstruction that I suggest you to look at if you have time. So there are some steps of the pre-processing that, for example, correct the fact that the slice time correction, correct the fact that we get images of entire brain every 2 seconds, but of course we get the scanner moves up over 2 seconds, so over 2 seconds we can get different sort of images, right, of the brain, of different slices of the brain. So we want to try to get all these slices on a common time point of reference. Then we have to correct for the subject movement. So each individual image is realigned to compensate for movement. And typically this is done by choosing a reference image. And then we have the issue of registering all the images basically to a common reference space, which is typically the structural image for a single subject. This goes under the name of co-registration. And then we want to make sure that we can compare individuals. So basically, it's typically, what they typically do is to align the data from different individuals into a common spatial framework, so that basically each brain of each individual is mapped to a common template brain, for example, using different types of atlases. And at the end of this process, we get to what we are typically going to use in our statistical analysis. So we have to be aware of this type of pre-processing. And this type of pre-processing can be quite huge. So this was a tweet like a year ago from a neuroscientist. So our raw fMRI data may actually be complex like this, but eventually we get cleaned fMRI data that are just a resemblance of the truth. This was a very provocative tweet. Well, to be honest, there is another way to see this. We can think about pre-processing as cleaning up the images. So when you take a photo with a camera, for example, there are several things you can do to make the image look better. For example, removing red eye, increase saturation. So in this regard, we may have a more positive perspective with saying that we are going to, our pre-processing is going to show the benefit or the signal of the data in a proper way. I think that pragmatically, our hope is simply that we are going basically to have a truth, and we are going to have a good approximation, I think, of the truth, and that's what we're going to be able to capture through through our analysis and through the pre-processing in particular. So the pre-processing is going to provide just a description of the big features of the data. So I don't have time to go into these, and probably I don't have also the expertise to go into the details, but I want to leave you with some additional resources. And in particular, there are a few books, chapter books. For example, this book by Paul Druck, Mumford and Nichols on Handbook of Functional MRI Data Analysis is a good one. Also, some chapters in the Handbook of Neuroimaging Analysis by Daniel Rowe and Strother and Churchill. If you want some web resource, John Michele webpage has actually a good tutorial in R to actually go through the pre-processing steps and learn the pre-processing steps yourself. This is something that I always suggest my students to go to. And also, if you want there are quite a few softwares that are used in the analysis of fMRI data. Standard software are typically FSL, AFNI, FreeSurfer, and SPM. And there is an online textbook and this brain book that provides you with the introduction to the use of a software, which allows also the pre-processing, to conduct the pre-processing steps. So I won't go further into the details, and let's see if there are any questions at the moment. So no questions yet. So I will continue with this quick overview of fMRI studies. And in particular, I want to point out now what are the major interests in fMRI studies. On one hand, one of the major objectives in the analysis of fMRI data is what goes under the name of activation detection, basically identifying which regions of the brain are activated by a certain task. For example, in response to an external stimulus, like seeing some particular type of pictures, as I was describing before. The other big type of interest is studying how the network functions of the brain, what goes under the name of connectivity. So basically how different brain regions interact with each other. And in particular here, the typical distinction is between functional connectivity, which studies undirected association between two or more MRI time series. You can think of these as simple correlation between the time series, or instead effective connectivity that studies how one brain region may influence another, the activity recorded in another brain region. So this goes a bit under the context, if you want, of causality, or anyway, how the activity in one region is directly influenced by activities in other regions. Of course, what one may use the fMRI data for is eventually to not only study the activity and connectivity of a brain per se, but also to try to relate this activity and connectivity to some particular psychological state, some disease state, or some behavior. For example, by combining multiple types of data and going into what are called integrative analysis. For example, think about the topic of imaging genetics, where brain imaging are used together with genetic features to predict some outcome of interest. Another possibility might be to use the fMRI data to try to identify imaging biomarkers. By imaging biomarkers, I intend some characteristic or regions of the brain that are whose behavior may be predictive, again, of some particular outcome. I'm thinking about, for example, schizophrenia, right? Maybe related, the onset of schizophrenia may be related with some particular characteristics of the brain activations, organization of the brain. So there is this quest basically to use fMRI data for eventually clinical purposes or in any way interventional purposes. So we will discuss mostly in this tutorial the first and second aim, if I have time, and in some cases we will hint upon the third aim, which is kind of a holy grail in many respects. And we will start in particular by looking at models for activation detection. We'll start by looking at single subject modeling. And here I will discuss basically the general paradigm by which typically the neuroscientists look at activation, how to detect activations of brain regions in response to a task, taking into account the spatio-temporal dependencies and trying to capture also functional and effective connectivity, that is how different regions interact. And take into account the fact that we are dealing with high dimensional volumes at each time point. After going through the single subject modeling, probably by the third hour or so, we will start discussing multi-subject modeling, and basically to try to see how to integrate information across subjects. What I'm thinking here is to describe multi-subject modeling as in a joint model. It's very easy to just replicate single subject analysis and combine them in some ways into a multi-subject framework. What I'm thinking here to describe are Bayesian models at that point that try to tackle the analysis of multi-subject data in a single model, or two-stage at most model, trying to basically describe at the same time the heterogeneity across individuals. But before we can go into the details of Bayesian models, we need to go a bit further into some aspects of fMRI data. And in particular, we need to come to terms with the fact that we are not really measuring neuronal activity at the deeper level, right? But what we are measuring is indeed the amount of blood that flows through an area. And it is true that we expect that when neurons in the brain become active, the amount of blood flowing through that area is increased. But at the same time, it is clear that what we are obtaining is a proxy of the underlying neuronal activity. So the signal measured in the fMRI data depends on the change in blood oxygenation due to the fact that a neuron is activated and basically some blood is sent to the area. And it is referred as the blood oxygenation level dependent or BOLD signal. But this also means that what we need to come to term is that we need to come to terms with the what is called the hemodynamic response function. Basically, the ball signal is going to increase at the onset of a task, but then is going to decrease over time after some time, basically, after 5-8 seconds, you're going to have a decrease after the peak in neural activity, and you're going to have an undershoot till you get to the baseline again, which is basically the state, the resting state, if you want, the state of no activity, basically. So, in other words, we have to come to terms that in response to a stimulus is not, our brain does not react immediately. If you show me a picture right now, it's going to take some small seconds, right? It's just some time in microseconds, right, for my for the blood, right, to reach the neurons, right, that are related, the areas that are related with the activation, right, with the response to the task. So the hemodynamic response function then is going to create some, we need to take into account this delayed onset of the blood oxygenation, right? When in response to a task, when considering the analysis. And this is a nice figure that I actually stole from Lindquist in a nice paper in 2009, where it basically describes the relationship between the neuronal activity and the ball response. In particular, the neural activity, for example, is characterized by a firing rate of the neurons, the onset latency, and the duration of activity. And this can be represented from the ball response in terms of magnitude of response, the time to peak of a ball response, and the response duration. And we have correspondingly some parameters in the hemodynamic function, the height, the time to peak, and the width that we need somehow to estimate. So that is why we need, in our model for fMRI data, to consider the signal from the ball response. And in particular, in the consideration of the signal of the ball response, we need to take into consideration the presence of this hemodynamic delay. One typical model that has been used in the literature to describe the activation in fMRI data is the model proposed by Princeton in 1994 that goes under the name of general linear model. I'm using the term general linear model here because this is the term that it is used in the neuroimaging literature. Everybody knows what you're talking about when you say general linear model, but this is not to be confused with the generalized linear model. Indeed, what I'm talking about here is simply a simple regression, by which basically I am regressing the observed fMRI data on the stimulus, but I'm thinking at the same time the stimulus function that describes the different tasks is indeed convolved with the hemodynamic response function. So, for example, in a block design where an individual is shown, let's say, one picture regularly over time, we are going to convolve the stimulus function with the hemodynamic response function that describes the delayed onset, and this is going to give rise to this basically convolved stimulus that describes the delay of the ball response with respect to the stimulus. And this basically is going to be our X matrix, design matrix in a regression setup. So in addition to the Ball signal, which is actually, if we want to study eventually how our brain responds to the different stimuli, right? Through the observations that we have obtained in the fMRI, right? fMRI measurements. In addition to the Ball signal that we want to capture, what regions of the brains are active, basically, we want to, we're also to take into account the noise, which may be due to hardware or physiological, due to the movement of individuals over in the scanner. So let me see if there are questions here. And I'm probably taking too much time here, but basically the basic idea here is, again, I'm having problems, sorry, with checking. I don't see questions at the moment. So the basic idea here, again, to try to somehow obtain models for the hemodynamic response function. In the slides, you're going to see different types of models. Typically, a model that is used is what is called the canonical hemodynamic response function, which basically is based on the linear combination of two gamma functions. And I'm not going much into the details here because I see that I am losing, I'm going a bit over the time that I had allotted for this. I just can comment that there are different types of hemodynamic response functions that are used. Some, in particular, the canonical hemodynamic response function has been shown to be quite good in capturing the behavior of a Ball response over time. And one important point is that the hemodynamic response function vary across the brain. That is, there are different areas of the brains may have different types of delay or peaks due to the activity. I'm not going to go into the details. There are a few resources where you can look at these problems. And at this issue, these are just a few of them, and also have put some more details in the slides that you have downloaded. I wanted to go a bit more into the statistical aspects, and in particular, first, I will discuss single subject inference for fMRI data, and I will in particular discuss first the frequentist approach. And as I said, I'm going to use the general linear model of Friston, and I'm going to in particular discuss first the general linear model for a single subject. And again, I call him general linear model because that's the literature. But in reality, the neuroscientist literature, but in reality, this is just a regression. So here, XV is the convolved design matrix, which is basically a matrix of T time points times P stimuli. And it's basically the convolved design matrix where we have a convolve the stimulus with the hemodynamic response function. We have our time series, which are collected at each voxel, at each point in the brain, at each regions in the brain. And these are basically time series collected at each voxel. And what we're interested in is actually determining how the design matrix, how the stimuli affect the changes that we see in the time series. And so the regression coefficient will capture the dependence of the Ball signal recorded at voxel V with the experimental stimulus for a given hemodynamic response function. And then we have an error term that we're going to discuss later, but basically these error terms can also consider the how hardware or physiological you can basically introduce into the error term dependence due to the also physiological reasons, to physiological nature of a task. So we will discuss that later on. So the main point is here, and we'll probably go fast on this, but it will take me probably 10 minutes, about 10 minutes to discuss. So the main point here will be to obtain inference on the regression coefficient. And typically, one may be interested in studying if the regression coefficient in response to a particular task, and for now, let's just consider a single stimulus, is going to be 0 or not. So, if there is going to be, basically, this is essentially a problem of variable selection in some way, right? Trying to understand if these the convolved stimulus is going to affect the activation, the time series, sorry, in that particular area. Mm-hmm. And the activation, detection activation is essentially trying to separate the different brain events in the different areas. So what is typically done in the fMRI literature? Well, I have a couple of questions. So I have one question that I will reply to later during the break. Another question instead is, what is YT? What is it measuring? So this question, so the YT is basically what you observe, are the time series that we observe in each, if you go back to the one of the first slides, right? When I had this, so basically YT is the time series recorded over each voxel in the brain. It is actually what we're measuring from the scanner. Does that answer the question? Great. If there are, if there is a need to clarify anything, please let me know. I feel a bit lonely here, not having the possibility to look at everybody, but have a typical exchange. Okay, so so we have, so as I said, so we are interested basically in detecting if there is activation in a brain when we use the general linear model, sorry. And this is basically a problem of hypothesis testing. So, and what it is typically done by neuroscientists is basically to fit a separate GLM in each voxel in order to obtain estimates of the coefficients in each voxel. And then they basically obtain basically maps of the coefficients or T-maps or p-values maps. These go under the name of statistical parametric maps in general. And in particular, often with SPM, I will refer in the future to maps of the beta nu hat coefficients. So by the inspection of these maps, one can identify regions where the activity where the Ball signal is higher in response to the stimulus. So, and typically what is done is to threshold these maps to find the voxels where the corresponding beta nu are significantly different from 0. Of course, we have to be aware of the fact that and this is a recent paper that I found quite interesting for the literature, right? Basically, there is, of course, a massive multiple comparison problem that we need to take into account. So failure to properly account for multiple comparison will ultimately lead to heightened risk for false positives and erroneous conclusions. And this is a picture that they showed to convince neuroscientists of the importance of multiple comparison. This is, of course, these are T-maps, and of course, by allowing a less stringent control, you are going to incur in a high likelihood of false positives and poor specificity. instead, you can decrease the false positives if you improve and get more stringent control. But of course, you're going to be risking a higher number of false negatives and smaller power. So all these are things that probably are known to many in the audience, but it's important, especially in the neuroscientist literature, to make the point of the importance of the of controlling for multiple comparison. And one way in which we can control for multiple comparison, which is often used, is family-wise error control. And typically, family-wise error control methods, they allow a stringent control over the false positives. And typically what it is used is Bonferroni methods or random field theory. Another option that it has been used recently is the false discovery rate, which is basically controls the proportion of false positives among all rejected texts. And the Benjamin and Ockper procedure in particular has been used a lot in this regard. So procedures that have been implemented to control the false discovery rate and allow an automatic and implicit threshold selection in neuroimaging. There has been, I'm going to conclude this section, and then I'm going to respond to some questions that I have received, but basically concluding that there is a heightened attention to the issue of multiple comparison in the neuroimaging literature. And in particular, this has come up recently in the discussion in the brain imaging literature due to the use of and the issues that Tom Nichols and others have found with regard to the use of a very popular procedure to obtain a cluster of activations that goes under the name of cluster extend base thresholding. Basically, this is a two-step procedure by which statistical significant clusters are determined based on the number of contiguous voxels where the test statistics exceeds some predetermined threshold. And the idea here is that there is a first threshold, which goes under the name of cluster defining threshold, that is used to threshold the voxels based on their amplitude and determines basically cluster of supra-thresholdy voxels. And then there is a second step where basically one considers the distribution of cluster sizes under the null hypothesis of no activation, and they basically decide which basically the significant cluster. So just as a simple cartoon, the orange line represents the uncorrected voxel piecewise over a row of voxels. So voxel-wise p-values over a row of voxels, right? This is the orange. You can think of these as p-values. There is a first threshold, which is the cluster defining threshold that defines the clusters. And then at the second level, basically, the significant clusters are obtained based in basically the cluster size as the statistics, right? So this cluster is deemed not significant, and this cluster is instead deemed significant. So this is a frequentist technique, of course, and it has come into focus recently that there are quite some pitfalls in the techniques. And this has come up in different ways from both the fMRI literature and fMRI neuroscientist investigators. And for example, advocating for a more stringent threshold of p-values, or instead also from statisticians that have looked at the assumptions underlying basically random field theory, and basically showing that the assumptions are probably not valid for the type of for the type of data that we have, and in particular, this paper that made in 2016 made a big how to say impact in the community. Basically, they show the parametric statistical methods are shown to be conservative for voxel-wise inference and invalid for cluster-wise inference. So in particular, the invalid cluster inferences are due to the spatial autocohesion functions that do not follow the assumed Gaussian shape. So this is just to say that there is indeed the understanding in the neuroscientist community that there is a need to take into account the multiple comparison problem. This has been tackled in the frequencies world in different ways, but there has been discussion, and there is ongoing discussion about what's the best way to proceed if you want. So for now, I'm going to stop here. Next, in the next hour, I'm going to take 10 minutes break, and I will actually try to answer the questions in these 10 minutes. So in the next hour, I will discuss Bayesian modeling of fMRI data. So 10 minutes, I will see you again at 10:11, okay? My time, so it's going to be so 10:11 Pacific time, US Pacific time, and I will come back at the time. In the meantime, I'm going to answer some questions. Okay, so the first question is, what I see is there no constant term in the regression? Well, there is a reference for baseline stimulus. So, for example, when the individual is in the scanner, the idea is that that's the situation of, let's say, rest. So that's typically taken as baseline. So that's what you use as your reference in the regression. Does that answer your question? Let me know. So the second question is, what is X? Have you observed X in some way? This actually is indeed a very good question. So X is, so we observe a stimulus. We do not observe the bold response. So we need, so basically when we convolve in the way that I described here, when we convolve the stimulus with the hemodynamic response function. So there are some parameters in the hemodynamic response functions that need to be estimated. So, so this is why I introduced actually this hemodynamic response function, because in the model that I will present, in particular later on, we will present model that I will allow the hemodynamic response function to be estimated. So it can actually, you could in principle, right, put in a value that is widely recognized as a good value to describe a good behavior of the hemodynamic response function, right? But it's best probably to estimate based on a parametric form, for example, of the hemodynamic response function. It is probably best to estimate the parameter itself. So in reality, we have not, so this is one challenge, right? But we have not really observed X. So we have observed a stimulus, but we have not observed a convolved stimulus. So another easy question to answer is this hat beta nu for a fixed time point. It's for each voxel. So I have so this is for each voxel, and it's basically we have y nu, which is a vector of time series of time points, right? We have xv that is also a matrix t times p. If you think about a single stimulus, this is a vector, right? T times 1 of stimuli. And beta nu is a single coefficient to describe, in this model, is a single coefficient to describe the activation in response to the stimulus. So it may be possible to think about that the vector or the coefficient might change over time, but this is not the case. It's simple a regression where you have for each voxel, right, t times 1 responses and one single covariate, let's say x, observed t times, right? Does that answer your question? Also, if you have further questions, I can also let you ask directly if you want. Just raise your hand, I think. Maybe you have a possibility. Let's see. Is it possible to control the t values as well as controlling for a decent effect size? I'm not getting the question. Do you want to maybe ask directly if you want?
Oh, hi.
Yes.
Do you hear me?
Yes.
Nice presentation. So often when I was doing fMRI studies, I realized that I can control for p-values. You know, sometimes I can find some regions that have like really really small p-values. That's because they are consistently small. You know, like the effect size is like consistently 0.0001 something. So, you know, that's like not predictive enough, which, you know, although you can say it's significant, but it's not predictive of the outcome. So I'm wondering if there is a commonly used technique that, you know, as well as controlling for the p-values, also controls for a decent, you know, threshold for the effect size.
Well, in the frequentist setup, as I see it, if I understand your question, basically, you're saying, okay, how can you, you know, if you, so basically, if you base your significance on the p-value, right? What's the possible, you know, so does that correspond to a real clinical significance? Basically, it's the usual question of a case. In the case, in that regard, this is actually a hard question to answer, especially in the frequentist setup. In the Bayesian setup, I will try. Maybe it may be possible, and that's probably one way in which you, I think, you may want to go to a Bayesian setup in this regard. If you think about some Bayesian variable selection techniques, which we'll not discuss much in detail today. I mean, we will discuss Bayesian variable selection techniques, but some of them are actually constructed so to allow the selection or encourage the selection of larger effect sizes. I'm thinking, for example, at the case of non-local priors, for example, that may discount small effect sizes, but promote larger or encourage the discovery of large effect sizes. Does it answer the question?
Oh, yes. Thank you so much.
Thank you. Other questions? If not, we still have 2 minutes before we start again. In the meantime, I'm going to remind you that you can look and download the slides directly from here. Oh, I had another question. So we have more than one stimuli at each time point. So we do have at each time point one stimulus only, but the issue that one may consider typically, right? I mean, we have just looking at one picture, right, at each time point. So, but one issue is that what we are actually have to take into account is the convolution, right, of the stimulus. So what we might have is that while the hemodynamic response function for one stimulus goes down, the one for the other stimulus is going up, right? So, in this regard, that's why we have that's another reason why we have the convolved stimulus. So the X is not simply a 0, 1 or 0, 1, 2, right, type of matrix, or right? Actually, it's not 0, 1, 2. It's a dummy, right? It's not dummy. It's variable. Mm-hmm. Does that answer your question? Let's see, perfect. Okay, so we are back 10:12, and we are back with the finally starting to discuss Bayesian methods. And well, one important point that typically comes up when discussing Bayesian methods is, of course, why should we go Bayesian, right? And luckily, I mean, the neuroscientist community, I found quite open to the use of Bayesian methods. This is also another reason why I would suggest you to look at this type of data and this type of problems in your own research, because the community is quite open as well in embracing Bayesian methods, at least to a certain extent. I think the reason is actually due to the work, and in particular these two seminal papers by Princeton in 2002. These were published in Neuroimage, which is one of the major journals for statistical analysis, especially of fMRI and EEG data. And the papers basically make the point about the use of Bayesian inference in neuroimaging, both from a theoretical and application perspective. And the reasoning of Princeton, which I'm going somehow to, of these papers, right, I'm going somehow to expound upon in this short course, is that the methods that we've seen quickly based on statistical parametric maps have actually a number of limitations. And well, these are things that we can all agree upon, I think, in this audience. So I'm not going to go much into the details, but basically, yeah, the p-value do not reflect the likelihood that the effect is present. So, and in this type of studies, it is more important and interesting to study the probability that a voxel is activated, or the probability that the activation was greater than some threshold. And this sort of inference is precluded, as we know, in a frequency setup. So what we would like to get is the probability of an effect given the data, and this is basically the setup typical of Bayesian inference. So if we were to rewrite the Bayesian, the generalized linear, general linear model, I'm too used to say to read GLM as generalized. The general linear model in the neuroscience literature as in a Bayesian framework, right? So, well, what we will do is just to consider the same structure as before, but basically have priors on the regression coefficients. And for example, setting up a mean prior mean value and a prior variance for the beta nu, and assuming, for example, normality. And of course, this is be a simple prior for the regression parameter. Notice that it is a p-dimensional prior, because we have basically a prior for each stimulus, right? So, and it is one prior for each voxel, right? So we have a specification for of each beta nu for each voxel. And well, if you do this is, I mean, if you assume that the error is also normal, this is basically simply the normal model, as we all know from Peter Hoff or BDA or other textbooks of Bayesian analysis. So we basically get simply that we are in a conjugate model, and the posterior distribution is conjugate to the normal likelihood. And so what we get is that so the prior is conjugate to the normal likelihood. I wrote this wrong. I apologize for that. I don't know what I was thinking, but anyhow, so the posterior distribution is still normal with updated variance, covariance metrics, and updated mean. And in particular, Friston and others suggest the use of non-informative priors in their paper. In particular, they consider the use of a zero mean and basically zero precision, right? So basically a flat prior. The posterior is proper, so in this regard, it is sort of okay. So, and this is a write-up from their paper. The Bayesian perspective is useful for minimum norm formulations because it motivates plausible forms for the constraints that can be interpreted in terms of priors. This is a point that I'm going to make later on. So one advantage of using a Bayesian framework in this context is indeed the possibility to introduce available information about the problem, in particular the fact that voxels are not independent of each other, but we may expect and take advantage of the spatial correlations. So, but even in this paper that was, let's say, one paper introducing basically the Bayesian formulation to the neuroscientists, and therefore it was made to be simple, understandable, and so not completely if you want, not meant for meant mostly to captivate the neuroscientists to the Bayesian literature, and so with that came with some limitations, if you want, from a Bayesian perspective. But the point was also, well, if you do this, then we are going to be able to obtain a fast inferential scheme, for example, by simple use of NEM algorithm. So this is I want to make this statement because, of course, when we go into the Bayesian world, in particular working with this type of data that I mentioned before are highly big data, right? There is high spatial resolution, and there is also high, I mean, these are still time series, lower time resolution than EEG, but still you have a sizable time series during an experiment. It is important to use computationally efficient methods, and therefore also EM algorithm or even approximate inference schemes are often fundamental or necessary in the study in implementing Bayesian methods for this data. And another point that this paper made, and again, think about the context, trying to allure people into the Bayesian world, and apparently it had a big impact in the neuroscientist literature. So is the following. So how do we take our decision once we have obtained our posterior of the parameters of interest? How do we identify regions of interest? And basically, the point that Princeton and others made was what probably we would come up with as Bayesian naturally. So basically, provide a spatial mapping of activated brain regions. Remember, in the frequentist literature, what we do is provide a spatial mapping of beta hat, right? So what we can do here similarly, but using instead the posterior probability maps, right? Based on the posterior beta of betas, right? And here, so I'm using their own terminology here. But basically what we have are posterior probability maps that are basically obtained by obtaining a posterior estimate, for example, the posterior mean of the model parameters at each voxel. Sorry, always spam. Okay, so we can obtain posterior estimates, for example, the posterior mean of the model parameters at each voxel of the brain, and basically map it to a brain template. And then once we have obtained the posterior means, then we can we can consider also the posterior probabilities well, we can map those, we can map posterior means, for example, then maybe we can try to find a threshold. And one possibility is to consider the threshold obtained by somehow by thresholding the posterior probability, let's say, of the exceedance region. That is the probability that beta, our coefficient, are going to be greater than, let's say, 0.1 or a given effect size. So this is the recommendation in this paper, and basically the choice of a threshold is going to be important in this case. But even you know, with this setup, right? I mean, I think a few points are made here. So first, we can use the posterior probability distributions to describe the inferences at each voxel. We can obtain, similarly as in the frequentist setup, a posterior probability maps, and we have naturally through the posterior distribution a way to take decisions and understanding and take decision and understanding which voxels, right, which betas, let's say, are relevant at the different voxels. So I'm going to go on 1 second more and basically show what this became basically sort of practice. And indeed, what an important point that I want to make is that this is this type of reasoning that by Princeton were also implemented in a software widely used by neuroscientists, a MATLAB plugin that goes under the name of SPM method. But here, so by SPM, here I mean the T, this is the frequentist, right? Setup, so in response to the frequentist maps, right, of T values, what we can obtain are posterior probability maps. And then the posterior probability maps represent basically, as I mentioned, for example, the posterior mean of the betas, right? And then there is a decision about which of these effects are relevant in a Bayesian setup by thresholding the posterior probability that the beta is greater than a certain value, basically by a thresholding of the posterior probability of the exceedance region. And so in this way, one can see that, you know, this somehow corresponds to a strict threshold. I mean, in frequency setup, what you'd have is a threshold on the T values, and one can appreciate the differences between the two methods, in particular with respect to you know, a more complete, if you want, a less stringent representation of the activations. And the comment here was one can appreciate how the pattern is similar but more complete than the activation profile inferred on the basis of the T maps. And so a few questions that I have received in chat. I will discuss, so there is one question that I will take later because it kind of anticipates things that I am going to discuss later. The question was, can you actually take into account the spatial consideration? Of course, and that's what basically the Bayesian modeling has done since, you know, these seminal paper, right? So the next model that I'm going to show are actually going to take into account the spatial aspects quite a bit. And then the second question was if one takes actually the precision equal to 0 in the prior, are we sure the posterior distribution is proper? We are in the normal model. So indeed, we are sure. I mean, this is the posterior distribution is proper in this case, but that's actually the question we should always ask if that is the case. And another question is, this is again a model fitted in each voxel. It is not a multivariate model. Well, correct. It is basically a Y V, which is basically observed at different time points, but it is the measurement at one time points. It is not a multivariate model in the definition of multivariate model that we've seen, but again, we will discuss extensions later on. And again, another student or another attendee asked, would there be any advantages or disadvantages in using a spike and prior distribution and inferring the activity voxels from the posterior spike probability? Well, you guys probably have followed yesterday's short course by Joe Antonelli and Antonio Linero, because you are definitely preceding what's going to come next. So I have I'm going slow here, but I hope that in the next half an hour, these questions will be solved. Okay, and yeah, another question was about the Gaussian mark random field prior that considers the correlation. That also is going to be discussed soon. So stay with me for a few minutes more. So I just wanted to conclude this by saying that So what we're going to consider is basically posterior probability maps. We can obtain, so I see that you guys are eager to move on to more advanced things, so I'll do that soon. Just let me finish a couple of things that I want to clarify. So first, we can easily in this model describe and compute posterior probability maps. And in particular, one point that I want to make is that posterior probability maps are not only a feature of something that can be used for fMRI data, but they can also use for other images, type of data, for example, PET images. Another point that I want to make, and I will make it fast, is that the rule that Friston made here, basically thresholding the posterior probability of this exceedance region, can actually be justified on solid, on more solid grounds by using basically Bayesian decision theory. The event beta nu greater than eta is what is called exceedance region of a spatial process. Basically studying where a process has values beyond, above a given threshold. So in particular, in the analysis of fMRI data, we aim to detect region-specific activations of the brain. Friston and Penny have described an empirical-based approach to build posterior probability maps of the signals, but what they suggest basically, and their approaches do not really rely on a optimality criterion that supports their decisions. But one could do a bit better in that regard, and in particular by setting up the problem into a decision theoretic framework. One can see the problem as simply, if you think at a simple model where we have a random field on a spatial domain S, described by two components, an error process, noise process, and unobserved random process mu S. What they're basically doing is testing if the unobserved random process is in an indifference region or not, right? So the exceedance region that we have on the beta, you can think of mu here as our beta. And basically the idea here is in a spatial decision theory framework, does the random field, so that does the random field belong to this indifference region in this location or not, right? So this is an hypothesis testing problem in the spatial setup, and there are tools that have been developed to tackle this problem from a decision theoretic perspective. And I'm not going to go into the details, but basically one can think of an indicator such that theta s is equal to 1 if mu s belongs to the alternative, or 0 otherwise. So this will be according to a true state of nature that we don't know. At the end of the day, what we need to take is a decision, either the process belongs to the alternative at this location, belongs to the exceedance region, exceedance interval or not. And in the case we decide that it belongs to the exceedance region, we're going to say that the null hypothesis is rejected. So there is so this tackles again and gets into a wide, vast literature on Bayesian decision theory. If you want a nice introduction about it, I would suggest this paper in Valencia Proceedings. I think it's available online by Peter Muller, Giovanni Parmigiani, and Ken Rice, about FDR and Bayesian multiple comparison rules. If you want to have a nice introduction to Bayesian decision theory, I think, I mean, in a paper, there is also a nice book by Giovanni Parmigiani and Lourdes Inoue, but probably that's too much, I think, if you just want to get a bit into the main concepts. I suggest you to look at this. So one point that I want to make is that we can actually at the end, what we can express the decision that we are making in these with posterior probability maps as defined by Princeton. We can basically describe these problems in a more formal setup, and we can obtain in particular a measure of the false discovery rate in expected in the Bayesian in the spatial setup by basically defining the false discovery rate as the expected value of the false discovery proportion that basically gives the number of false positives over the number of rejections, right? And there are also other measures, but basically by considering basically we can control the false discovery rate by developing and considering a decision theoretic framework, in particular by considering a compound decision theory framework, by which we consider a loss function that compounds the number of false positives with the number of false negatives. If we do this, and we set the problem in a Bayesian decision theoretic framework, the original suggestion by Princeton actually comes up to be quite natural, and in particular to be the optimal suggestion. In particular, one can show that the decision rule that minimizes the posterior expected loss is of this type, where basically we have a threshold on the posterior distribution of the alternative hypothesis. So the probability to reject given the data, the probability that theta s is equal to 1 given the data, the posterior probability of alternative. If we can obtain an estimate of this quantity from our Bayesian procedures, and by thresholding the posterior distribution of the alternative, we actually are going to obtain the decision rule that minimizes the posterior expected loss for given penalty lambda. So, in other words, the suggestion by Princeton actually was a good one in that regard, because it minimized the posterior expected loss. So it turned out to be if you want, the right thing to do, assuming this loss function, of course. And the good thing about this is that actually we can obtain, so all of these, of course, is dependent on a fixed value of lambda. That is given lambda, given a certain penalty on the loss function. If you're familiar with the lasso method, you can think about this lambda as sort of the same thing, right? Giving a penalty on the number of false positives in this case. But here, this rule is obtained with a given lambda, but at the same time, what we can do is, since this is something that you can, so we can compute the posterior probability of alternative, is basically to use tools that have been actually developed by Newton and others in the context of genomics, for example, and estimate basically, come to with a measure of Bayesian FDR and estimate the false discovery rate at each location by basically considering the posterior probability of the alternative and basically this quantity here. So we can estimate the posterior probability of the alternative through MCMC iteration. We can do this for each voxel rule. And basically we can choose and this threshold K is basically this 1 over plus lambda, right? So for given lambda, we get a threshold kappa, kappa 1 over 1 plus lambda for given lambda. But so if we want to control the false discovery rate at 0.05, what we can just do is simply have an equation where what we have here on the right side, which can be computed simply through MCMC, is going to be equated to 0.05. So this is going to be by reverse engineering, we're going to be able basically to obtain the value kappa that allows us to obtain a false discovery rate of 0.05. This is all to say that the threshold, we can threshold the possible probability of alternative, like Princeton was suggesting, but also we can choose a value of a threshold in a more thoughtful way, if you want, in a more principled way, by using this Bayesian FDR estimate and basically reverse engineering the estimates in order to find the optimal, let's say, the value of kappa that is going to allow a false discovery rate of 0.05. Does that clarify or helps? I mean, are there any questions about this? So this is all based on first principles. And yes, so in other words, what Princeton was suggesting was actually, as we were sure, I mean, it was coming directly from the Bayesian thinking, right? But it has also solid underpinnings in the Bayesian decision theory literature. Okay, if this is okay, and I don't see anyone screaming in the chat at the moment, or about something that I may said or something that was not clear. So what we can do is move on and actually see more, let's see, more complex. I don't see the chat anymore, but I think I can easier. It is easier if you guys write in the Q&A, because I can see it more prominently for some reason. I don't know. Okay, so in terms of okay, so the question, yeah, I wasn't the idea when I was saying, so the question is, if you force FDR to be 0.05, aren't you risking the losing control of power? You got an imprecision that I said while talking. The idea here is to have basically to find, so the control of 0.05 is your you want to control false discovery rate to 0.05 at this level, right? So, but what you can do is only at this point decide which ones, right, are going to be which decisions are going to which voxels are going to be decided in favor of alternative versus the null. And so the procedure that I was describing to set to 0.05 was the procedure to get the kappa in the existing data set. The fact that by doing this, actually you control the false discovery rate is indeed due to the fact that a repeated experiment, right, the nominal FDR is controlled, right? So does that answer your question? I didn't go as much into the details. On many things here, I won't be able to go much into the details. I want to give the basic ideas, and I hope that you guys can go back also and look at the slides later on with, you know, and look at the details in more specificity. But thanks, yeah, it's good. Yeah, it's okay. So see that time is running out again, and here we are getting to the important and interesting stuff, basically incorporating prior information into the model. So we want to, in particular, take into account spatial and temporal dependence for this type of models. The spatial dependence is indeed something that we should expect in this type of analysis because of the fact that the response at a particular voxels is likely to be similar to the responses at neighboring voxels. So in the way in which typically spatial dependence is captured in Bayesian models is by imposing spatial priors on the model parameters. And here we can think about the typical ones, for example, Gaussian mark-random field priors, conditional autoregressive priors, Gaussian process priors, and what we will also discuss are spatial variable selection priors. And so there is a wide literature, and in the spirit of this short course, what I want to do is somehow provide a sort of a reason, probably biased because it's based on my likings as well, or my understanding of the evolution of the literature in this regard, but a sort of understanding of what has been done in the literature and the points that have been made also with respect to the work with neuroscientists in some ways. So one obvious way to introduce spatial dependence is by using a Gaussian mark-random field prior on the regression coefficients beta, and this is for each stimuli, right, and each voxel, we can think that we have a vector of coefficients of dimension v times 1, the number of voxels being v. And many authors have used a Gaussian mark-random field prior specification for describing the dependencies across the betas. And basically the idea here is that the precision matrix Q has basically structured as a particular structure by which on the diagonal there are on the diagonal for each voxel there is the number of neighbors of each voxel, and then there is -1 anytime a voxel is neighboring another one. But other than that, the precision matrix is very sparse, because if the voxels are not neighboring, then the other terms of the precision matrix are 0. So this is a very simple way to introduce a spatial dependence, and basically this is equivalent basically to assume that the prior for beta has this form, which basically results in this conditional representation by which the voxel, the distribution of the beta coefficient for voxel v is related to what is conditional on the value of the voxels in neighboring regions. And in particular, you can see that the assumption is that the normal, the conditional distribution of the betas at voxel v is a normal with mean, prior mean, that is basically the average of the values of the coefficients in neighboring regions, in neighboring voxels. So the activity in a voxel basically is estimated by the response magnitude in the voxel as well as in neighboring voxels. This somehow creates smoothness, right, of the activations. And the idea that underlies this sort of models is that the activity, the expectation that the activity happens in regions, so it is not isolated in single voxels. So the idea of independent activations is not optimal. One issue with this is that the matrix Q is in less than full rank, so it's improper, but this allows basically to avoid specifying a prior for the mean level of beta, and also the intrinsic Gaussian mark-random field prior that is used here gives the maximum correlation possible if one uses a first order neighborhood. So I'm not going to be able to go into the details of Gaussian mark-random field priors. So there are great monographies by Banerjee, Carlin, and Gelfand, and Roux and Held that can give you more information about Gaussian mark-random field prior. Somehow related is also the idea of Laplacian prior that has been proposed by Princeton, so by Penny, Trujillo, Barreto, and Princeton in a paper in Neuroimage. And in this case, again, the idea is somehow to take into account the spatial dependence between the betas, which is informed by the fact that voxels may be neighboring to each other, may be close to each other. And in particular, what they consider was instead of the Gaussian-Markov representation that we've seen before, a very similar one, where basically they use a spatial kernel matrix, but in this case, what they consider is a Laplacian operator in particular, that basically makes this again, it's based again on the idea, on the structure. So the Laplacian operator is such, the element of the Laplacian operator is equal to 1 on the diagonal, is equal to -1 if the two voxels are adjacent to each other, or 0 otherwise. And one good thing about this is that one can then consider simply basically obtain this prior, right, by simply considering a random variable omega distributed as a normal with variance alpha j, and then basically by virtue of Laplace transformation basically obtain beta j as L-1 wj. And it is very fast to obtain. It is very easy, so much that actually this is one of the spatial priors that are implemented in the software SPM, which is the software. It is basically a MATLAB plugin that was pushed forward by Princeton and Penny, and from the Wellcome Trust, and basically it is one of the tools that have been historically used by also by neuroscientists to conduct this analysis. So in this regard, this is also one of the reasons why it may be good to, I mean, I wanted to show it because this idea is indeed implemented in a software widely used by neuroscientists. Or at least that has been in the past widely used, probably not as much today. There are other softwares, but it is known for sure. And in particular here, I want to point out the effect of using these spatial priors. So in particular, as I said, these priors enforce smoothness by penalizing the differences between neighboring voxels. And it is also a prior that is used not only in fMRI, but also for EEG data. Let's see a bit in a simple simulation where they did the effect of the prior. In particular, they consider in their paper the true regression coefficient with the true effects. Then they use an uninformative prior that can be obtained by simply considering S equal to identity, right, and taking alpha small in the previous presentation here. Then we consider a G prior, that we call a global shrinkage prior in this paper, and then we consider a Laplacian prior. And basically the idea here is that the Laplacian prior is able a bit to reconstruct the accuracy, right, or with some accuracy, the true regression coefficients by decreasing somehow the noise all around, right, and all the big chunk of false false findings, right, that so basically it's good somehow to denoise or to smooth out the signal in order to see exactly where the regions, right, of higher effect, let's say, of higher effect size somehow, right? Good. Okay, so, and same thing also on real data, we sort of idea here. So one point that also they mentioned is that, yeah, somehow when apply to real data, we see somehow the same structure, but at the same time, Laplacian prior results in a much higher estimates of effect sizes than other regions. And I'm seeing that I am going a bit behind again, but let's see if there are questions. I'm always overambitious. I may cut some slides from time to time just to go to the meat. So there have been quite some works that I have actually put here on the slides for you that you may want to look at that extend the basic ideas. So for example, Harrison and others use a weighted Laplacian prior that have weights that depend on the distance between voxels. Instead of using a Gaussian Markov prior, for example, Derardo, Dubois, Bowman, and Liu Zhang use a multivariate car. I'm also putting these papers here for you to look at so that you can look at them later, but also understand and have a bit of a feeling if you're not part of this field, of the type of the people that are working here for in this domain, right? For example, Bowman, he's someone that has done a lot for statistical imaging, so definitely someone to look at. I wanted to just point out also to another extension in this case, by Liu, Berrocal, Barsh, and Timothy Johnson. This was basically the use of fMRI data to for a different purpose than usual. So in this case, the use was using fMRI data for pre-surgical studies. Basically, the idea here is to understand what regions of the brain should be targeted in a surgery. So this is a bit different than the typical problem that we have of activation, right? Using fMRI data to detect activation. Here, the idea is to try to detect regions of the brain that should be, for example, the target of some surgical procedure or excised or anyway looked after. So here the point is indeed that we need to be careful that the type of the type of loss function that we want to consider in our decision problem is a bit different, because in this case, actually what we are more interested is false negatives, right? The more than false positives. In the sense, to a certain extent, of course, but in the sense that if we are not able to detect areas that should be targeted, then this is going to be more costly and in the long run, and then possibly detecting areas that, or parts that we should we should have not targeted. So in this case, false negatives are more dangerous than false positives. So what they did was basically to propose a spatially adaptive CAR models to map functionally relevant brain regions that could have in pre-surgical evaluation of this tumor resection. So they consider the images, which are contrast summary images of the time. So we don't have a time here, but they have voxels. And basically, in their proposal is simply to use a Gaussian Markov field formulation, or actually a CAR model. But in particular, they consider not only a CAR model on the means, but also on the variances. So in particular, so this allows basically somehow the CAR, the variances to change, right, in the different locations. So this is somehow sort of using at the same time not only the level, but also the it's similar to if you're familiar with the let's say shrinkage priors, right? Like, for example, the horseshoe prior, right? Using the variance, basically a scale normal of Gaussians, right? To determine which regions are important. So there is not only a spatially invariant process for the mean, but a spatially invariant process for the variances. And long story short, and again, I have, due to time, the necessity to sort of skip on some details, but I hope that I was able to give the basic details, right? And the idea here is that one wants to be able to obtain a better representation of the areas, and in particular, you want to obtain at the same time, right? Areas that are comprehensive, right, but not as much. So in some simulation they show, and in some application to real data, they showed that their method performs well. Their method is a CWAS in this case. And a few other points that I want to make just to conclude this second hour, and it's the paper by Tang, Natu, and Johnson. Again, they basically use, again, a sort of same ideas. They use the Penny's prior for spatial modeling, but also they use a Spikens prior to model the coefficients of the air process that governs the temporal nodes. We will go back to these type of priors later on. To the Spikens lab. I just want to point these here just because it's related somehow to this model by Penny. Okay, so before we continue, so this is the second hour, and I need a couple of minutes of break as well. So in the third hour, we will finish up the discussion of the activation for single subject. And in particular, I will discuss this other nice paper that has been recently proposed by Per Sieden, Anders Hägglund, David Bolin, and Matthias Villani. And then I'm going to discuss quotient process priors and spatial variable selection priors as well, hoping to be able to provide a general presentation of the models for single subject. If there are any questions, please feel free to add them to the chat, and I will probably go. Okay, yes, there is a question here by, go ahead. Zanzwan? Oh, sorry. I was trying to, can you accept the, can you talk? Or, okay, let's do this anyway. So let me see if I can do this again. And let's meet anyway at 11:12 in case, so in 10 minutes, in case you guys want to go, we can be back in 10 minutes. John Swan, apparently I cannot have you unmute for some reason. If you could write the chat in the chat, and I'm gonna be back in a few seconds. In a minute.
Hi, Professor, you are muted.
Okay, if there are any questions, please feel free to ask. If anybody can talk, but actually gives me the possibility to relax a sec. So thanks for that. If you want to chat. And I'm making some coffee just in case. I need why is it? Okay, let's see if I can do this way. Nope. This should be okay. Is the pace okay? Oh, okay, thanks. Okay. Feel free to scream if there is anything that I should discuss more in detail. We're going to be together for 3 hours, not 3 years. Just in case, remember, you can download the slides. So even if I had to, I would probably have to skip something or to jump through some hoops to be able to do everything. But you have full slides here. And as you probably have noticed, I'm not saying anything really that different. Perfect, is 11:12 Pacific. So thanks for sticking with me. And the next block, I'm going to discuss again ways to incorporate the prior in the Bayesian setup. And in particular, I will also make some consideration about you know, as you've probably seen, what I'm going to do is probably also show you different type of papers that have been presented or published recently in the literature, so that I think, I mean, they can give you a good understanding of what's out there and what things might be improved, what are the sticky points, let's say, that one can think about. So in particular, I wanted to point out to your attention this paper by Percy Den, Anders Eklund, David Bolin, and Matthias Villani. And one thing that they do is whole brain fMRI analysis. An important point that we need to be aware of, again, is the fact that we are dealing with big data. And often what is done in the analysis of these data is somehow to take some dimension reduction shortcut, for example, by looking instead of an entire brain at some slices. We will see some examples later. Let's say some slices of the brain instead of the entire brain. Another way that it is popular is, instead of considering all the voxels, to consider some regions of interest, or divide the brain into sets of regions, for example, by considering the mapping of the different voxels to a particular atlas. So doing a whole brain analysis of fMRI data is, of course, challenging from a Bayesian perspective, from a computational point of view. But there are a few people, in particular Matthias Villani group, that have worked quite a bit in this regard. And this is a nice paper that was published 4 years ago, where basically they consider basically a whole brain Bayesian methods. In particular, as you can see here, their motivation is that a popular method, Bayesian methods, is the one that is standard in the SPM software, and it is the one that I have described before by Penny and others. And the method processes the data slice by slice, and then uses an approximate variational-based estimation algorithm to actually, in the software, obtaining the inference. They actually, in this paper, they actually consider a fast MCMC scheme for exact inference in the same model, both slice-wise and for the whole brain, using a 3D prior on activity coefficients. And the idea here is basically quite, and the main contribution of this paper is also from the computational standpoint. So we start with a model in Penny that is written as follows. I think it's 2005, sorry. And again, it's the model that we've seen before, just written in a matrix form in this case. And each coefficient is assigned a spatial prior, like we've seen before. I called before with S transpose S, if you remember. And this is now, I'm just calling D. In this case, this is simply a fixed spatial n by n precision matrix. It is fixed. This is important part, and it is equal to -1 if I and j are adjacent, and 0 otherwise, except on the diagonal. And this can be basically rewritten as we've seen before, basically in terms of only of the alpha. But if their point here is they can obtain the conditional posterior for the vector of the coefficients in the following way, right? And this is basically the expression that they have to use basically this can be recognized first basically as the kernel of a normal distribution with mean omega tilde -1 omega w here, and then with precision omega tilde -1. Where this omega tilde is indeed quite complex, right? And this characterizes a computational bottleneck, because in order to obtain the mean, for example, one has to consider and obtain the inverse of omega tilde. So this paper is basically, in this paper, basically obtain an algorithm to obtain omega tilde, to obtain the inverse of omega tilde. And we have to present two algorithms, I have to say. The first algorithm uses the Koleski decomposition, and then reorder the rows and columns of omega tilde, such that also the triangular matrices of the Koleski decomposition will be sparse. The second algorithm instead uses a method by Papandreou and Yuli to avoid the Koleski decomposition, and it is basically based on rewriting the prior in the following way. By considering the differences between adjacent boxes, which are ILD normal, and basically by considering then a conjugate gradient method to obtain an approximation of the mean. But this is just to say, more than for the details, that one has to be somehow creative if they want to actually do a full-scale analysis of fMRI data. So computational issues are going to come up over and over. So if you think about this, we are still looking so far at the single subject modeling. We still have to look at multi-subjects, right? So we want to take into account all the information from the data, but this comes, of course, with some trade-off. Another alternative type of priors that one can consider are Gaussian process priors on the coefficients beta. And in this case, the paper that one can consider is the paper by Harrison and Green, 2010. This is the first paper that proposed this type of, they use the Gaussian process priors for beta coefficients. And the way in which they do it is, again, quite simple due to the computational aspects, right? So basically, they simply consider a simple Gaussian process prior with a basically exponential covariance function, to the Gaussian covariance function. An important point that I want to make here, but when you look at the Gaussian process priors, that it is probably goes unnoticed, is that basically one is thinking instead of the area grid that is characterizing the brain, basically the process, the support of a process is indeed a continuous random field, right? So there is this added assumption, right, that the observations are basically taken from a continuous random field instead of the area grid, which is typical of the instead of the data itself, right? But at the same time, it of course provides greater flexibility, and it gives the possibility to make predictions also at locations where measurements may not have been taken for some reason. But the model is very simple. So basically, they just consider a simple covariance structure that is based on the modulus of the distance between two voxels. And they use basically a variational-based approach to obtain inference in this case. And there is a question. Oh, this is a great question. So the question that I got is, from a neuroscientific perspective, do you think that imposing sparsity is justified? Aren't we at risk of missing complex patterns? Yeah, it's a good question. So there is this idea that, and we will get back to this, that typically activity is coming into clusters, right? So definitely some areas of the brain, I mean, this is sort of somehow accepted in the neuroscientific literature, that one should somehow identify sparse regions of activations. But indeed, so on one hand, I mean, if there are complex patterns, we should probably try to have priors, right, that allow the possibility to describe these complex patterns as well, right? At the same time, enforcing sparsity if needed, right? Or if indeed we have a sparse structure, right? All these priors, indeed, I mean, we do not. I mean, they take into account the spatial information, but the idea of sparsity itself is not. I mean, okay, especially I'm thinking about Gaussian process priors in this case, right? There are some assumptions, but in typical, I mean, they shouldn't. Let's say that we believe in sparsity, so we are encouraging sparsity, but in principle, I think that all these priors allow a possibility to obtain. It's not, let's say, a hard selection or hard sparsity. I don't know if it is clear what I'm trying to get at, right? So, but it is a great question. So on one hand, just to answer your question more directly, I would say, yeah, there is indeed the idea in the neuroscientists that the patterns of activations are maybe complex, but somehow localized, right? What I described probably at the beginning quite fast was this idea that activation tries to understand the organization of the brain, understand the separation of the different brain regions and the activity of the different brain regions. So there is this sort of idea. Now, as I mentioned before, in addition to studying activation, there is also the study of connectivity, right? And in that regard, there is indeed a big debate if, I mean, in terms of networks, right? If the idea of small networks, which is somehow the one prevailing in the literature, is actually the appropriate one. But the complex organization of the brain, right, of the complex ways in which we answers to task or response to task can be captured, I think, in the combined analysis of activation patterns together with connectivity patterns, right? So it is how brain regions work together in response to a task. Did I answer your question, or just I messed up more? Or there is a follow-up question? If there is a follow-up question, please. Also, you can always raise your hand. We can try again and have you talk so that I can rest my voice for some seconds. Good. Okay, other questions? Nope. So let me move on. And I believe that many of you have seen yesterday a nice short course, actually, very good short course on variable selection, Bayesian variable selection. So many of the things that I'm going to discuss right now are probably going to be familiar with you or reminiscent of things that Antonio and Joseph have discussed yesterday. So the idea here is indeed that what we are studying or trying to understand is if coefficients are significantly or relevantly different than 0 at different voxels, right? So is there an activation or not at the different voxels? This is essentially a problem of variable selection, if you think about it, right? Understanding if beta at the different voxels are 0 or not. So a large type of priors that have been used in the scientific literature is indeed represented by spatial variable selection priors. And the idea here is, again, taking the standard general linear model by Princeton, basically identifying the non-zero betas. And so we can do these in different ways, and probably after yesterday's short course, you can probably teach me many more ways to do it. But one classical way to do this put this classical variable selection problem in a Bayesian setup is by using a mixture prior on the beta, a spike and slab prior on the betas. In particular, this is a prior that somehow selects activated voxels. So if a voxel is not activated, then let's be gamma nu an indicator variable that is 0 when the voxel is not activated, and 1 when the voxel is activated. If beta nu is if the voxel is activated, the indicator variable gamma nu is going to be set to 0, and beta nu is also set to 0. If the voxel is instead activated, the indicator variable gamma that identifies if the voxel is activated is a latent variable that identifies the voxel is latent binary variable that identifies if a voxel is activated or not. If the voxel is activated, gamma nu is set equal to 1, and beta nu is taken from a normal distribution with large variance tau. So the spike and slab prior is widely used in the Bayesian setup to describe activations, and Antonino Linero and Joseph Antonelli described these yesterday. So to connect these with the discussion that we had before about the selection and posterior decisions, of course, our problem now will become to understand if the probability that gamma nu is equal to 1, the probability of the alternative distribution, right? The probability that gamma nu is equal to 1 a posteriori after seeing the data is greater than a given threshold. So basically what I mean here is that what we are going to monitor is from our MCMC, the average of the gammas, right, to approximate the posterior probability of the alternative, that is the posterior probability that gamma nu is equal to 1. So with spike and slab priors, the inference is based on what goes on thresholding the posterior probability of inclusions, that is the posterior probability that gamma nu is equal to 1 given the data. And we're going to set up this up in a decision theoretic approach as we have discussed previously. We can approximate the marginal posterior probability of inclusion by computing the proportion that gamma nu is equal to 1 across all iterations after burn-in. And again, we can obtain an estimated activation map, basically, by selecting all voxels that have a posterior probability of inclusion greater than a threshold value. And if you remember our discussion before about the Bayesian false discovery rate, what we can do is basically select this threshold in order to control the false discovery rate. For the given value of the estimated false discovery rate, right? So this basically is a simple, straightforward setup by which we are able to obtain basically which voxels are activated as a result of the application of the use of a spike and slab prior. And so the point now is try to model the probability, the prior probabilities for gamma nu, right? What prior distribution we're going to assume for gamma nu. And this is what has been done in the literature in different ways. For example, the first example is a paper by Kalus, Salman, and Farmeyer that specified a spatial probability model for the probability of activation. And basically the idea is to assume that a probability model for the probability that gamma nu is equal to 1, where the there is a latent random variable alpha nu defined in each voxels that basically is an element of a v-dimensional vector that basically follows a Gaussian Markov-random field prior, or intrinsic Gaussian Markov-random field prior, as you've seen before. So in a way, the idea is to use an underlying latent process, this alpha nu, to define the dependencies across space and basically influence or determine the probabilities of activations directly, right? The probability of activations. Other questions? Please, and again, also please free to raise your hands. I should be able to see. The other option is instead to use different models. For example, a model that uses the fact that gamma nus are 0 and 1s. For example, directly a Markov-random field prior on the voxel-dependent indicators for activation. The Markov-random field prior is basically a prior that links the probability of activation at the voxel nu to the voxels nearby, basically to the activations in voxels nearby. And here, the basic idea is the following. So if you look at these characterization of a prior probability for gamma nu equal to 1, this is basically the results of two components. A component that it is characterized with this parameter d, they control the sparsity, basically without considering the neighbors, right? For a second, think as this component is not there. The parameter d is basically a sparsity parameter that controls the expected number of activated voxels independently of the neighbors. Basically, this is just a, if you want, base prior probability of activation. This prior probability of activation is going to be increased if other voxels are also nearby, are also active. And the magnitude of increase is regulated by this parameter E that is basically is a smoothing parameter that affects the probability of identifying a voxel as active according to activation of the neighbors. And there have been quite a few papers that have considered similar priors before and together, right, in this similar type of priors in the for modeling the activation gamma nu. For example, Smith and Meyer 2007 is another very good references, and Lee and others. They considered similar priors also to incorporate anatomical information by modeling the gamma nu. Another point, so questions so far? Any, again, feel free to raise your hands and ask. Of course, so far we have been looking at spatial dependence. Of course, we have also to consider the temporal modeling. And here, so the fact that actually these are time series, right? So we assume that there are going to be temporal dependencies. And typically the dependencies are taken into account in two ways. On one hand, there is temporal dependence that is naturally created by the hemodynamic response function. The hemodynamic response function describes, if you remember, the delayed onset of the response due to the stimulus, right? And this is somehow can be used also to capture the temporal correlation between different time points in some ways, right? It's kind of naturally implicit. In addition, the temporal correlation can be captured through the modeling of a noise component, this epsilon nu. And so far we're considering epsilon nu basically, except in a simple case where I sort of noticed that there was an autoregressive process on there. I basically assume so far epsilon nu as independent terms, but typically one may think that there may be some sort of, you can see the epsilon also as time series, right? And you can see, for example, you can think about having a autoregressive structure of some type on the epsilons. And there is quite an extensive literature on this regard. Another different way to see the process is instead of considering autoregressive processes, is instead to consider and trying to model long-range dependencies in the fMRI time series. So there is this idea that basically fMRI time series behave somehow like fractal time series. This is what Bulmer wrote in, for example, his paper 2004. And basically they exhibit long-range autocorrelations, long memory in time. And one possibility then is to try to obtain and to model the matrix sigma nu, which is a T by T matrix in this case, by considering terms, gamma elements, gamma ij, that, or sigma ij in this case, because as a function basically of a term that basically, of terms that basically model long-range dependencies, right? So there is this innovation term psi, and there is a parameter alpha that defines the decay of the dependence at different times, and basically it's a decay parameter that basically describes the long-range autocorrelation of the time series. So the issue with this representation, however, is that if we do this, we are basically bound to a structure for sigma, which is full, right? So, and again, we do not want to work necessarily with full matrices, full variance covariance matrices, because from a computational perspective, this is going to be quite tasking, right? Quite daunting. So we may need, if we use this type of approach, then to somehow get to a simpler variance covariance matrices. And one way to do that is to use wave transform on the original data to somehow widen the data. And so if we use the wavelets transform, then we basically obtain a new data set that is but basically decorrelate basically our long memory processes. What we have is a new GLM, basically general linear model, with new data obtained through the wavelet transform. And the important point here is that the error term here is characterized by this matrix sigma V star, which is basically essentially a diagonal matrix, where the term on the diagonal may be complex, but depend on the two parameters, psi, the innovation parameter, and alpha, the long memory parameter, right? I forgot to say that alpha here has to be between 0 and 1. So, but basically by using the Wave transformation in this case, Wave transform in this case, we get a model which has decorrelated noises, basically. So, except, I mean, possibly complicated function of the diagonal, the variance covariance function is greatly simplified and basically essentially diagonal for all purposes. So, and that is definitely something that we want to exploit and makes of course, computations much easier. Questions? Okay, so I still don't see questions. Okay, so now the point at this, it will be then to add a prior, right, on psi nu and alpha nu. And one way in which the priors can be added, right, is, for example, some type of priors. Maybe, well, this is my second love, if you want. So one possibility is to try to induce some sort of clustering between the parameters alpha nu and psi nu. Notice that if we induce clustering between alpha nu and psi nu, basically what we're saying is that we're clustering time series characterized by the same type of temporal correlation, right? And this is basically sort of the idea behind functional connectivity, identifying basically voxels characterized by the same sort of behavior of a time series, right? So one possibility then is to use a cluster-inducing prior, and I'm using this in a loose term, but basically a Dirichlet process priors on the parameters of a long memory process. Now, I'm saying cluster-inducing priors with some caveats. Basically, the idea here is simply to group somehow the psi nu and alpha nu in groups of voxels behaving somehow similarly from a temporal perspective. More on Dirichlet process priors and Bayesian parametric priors will be given tomorrow, right? More details in the short course by Thanos, Thanasis, Cottas, right? So I'm not going to go into the details here, or I'm just saying that what we were trying to do in this case was simply to induce clustering of the fMRI time series. The idea is somehow allowing automatic grouping of the parameters with data-driven number of clusters. And of course, as Thanos will describe tomorrow in length, one has to be careful about how they discuss about clustering with respect to the partitions induced by the Dirichlet process. But so far, for our application, this was okay. So for details on VMP approaches, follow the tutorial by Cottas tomorrow, if you can. Another, if you cannot, another possibility might be just to look at these references. There is a nice book by Peter Mueller and Fernando Quintana, Bayesian parametric data analysis. Probably also Leandro Hara, he is in the book. I apologize for not adding him. And another book that it is a bit more mathematically mathematically rigorous, let's say, is Fundamentals of Non-Parametric Based Inference by Subhashis Ghoshal and R. van der Waart. And I suggest you to look at these among many others. But definitely a good starting point is this book by Peter. Here, I just wanted to show you an example, since I never showed examples so far much. And in particular, this example was on data obtained from the Welcome Trust. Here we had a single subject for different conditions. So we were looking at different organizations of points on a screen. And just to give an example, right, we just consider a stimulus function, which is very easy, one coded as 1 for the case of there is attention or no attention, steps instead so basically anything other than fixation is coded as 1 and 0 is coded as fixation. And we look in particular at 3 different slices. So the structural MRI images for these slices are as such. So this is the posterior activation map that we can obtain by looking at our MCMC. And this is the posterior activation map using SPM8, which is if you remember, SPM8 implements the penny, the software, the penny method, right? So in particular, you can see that by considering the variable selection prior, in this case, with spatial variable selection prior, you can get a more sparse representation of the activations. And also, of course, the temporal aspect, the way in which we, in this case, we consider the long memory dependence also had importance. Here, posterior, I'm just showing posterior mean maps of the betas, and this is the posterior mean maps of lambda, where lambda is in this model, I didn't discuss this, is basically a parameter of the hemodynamic response function that is allowed to change over space. So as you can see, this is basically saying the hemodynamic response function indeed changes over space, and in particular, you can see also changes in the regions that are deemed as activated by our method. We also had this spatial derivative process for alpha nu and psi nu for the innovation and delay parameter of the DP, and these are the posterior clustering maps obtained from the derivative process with the posterior mean maps of psi and the posterior mean maps of alpha. And the innovation parameter and the delay parameter, they're very difficult to interpret like this, but if one goes into the details, one can see that the active voxels are characterized by typically by lower variance and increased autocorrelation, whereas lower variance and increased correlation for the active voxels, whereas instead the non-active voxels are typically characterized by an increased noise and less dependence over time, which is actually what you would expect. So, questions? I went a bit fast, but if not, I wanted to point out to you these 2 other papers. One is a nice paper by Chen Han Yu, Raquel Prado, Hernando Mbao, and Daniel Rowe. It's a very recent paper, 2018. And basically, they work and they made, so basically, their starting point is that when you get data from the scanner, they are eventually transformed into basically real objects. But indeed, the voxel time courses from fMRI data are complex value signals that give rise to both magnitude and phase data. So, so basically they discuss and they basically have methods to detect presentation from complex value fMRI data. Basically, we adapt the type of method that we've seen so far to deal with complex valued fMRI data. It's a nice paper that I suggested to look at. And this is instead a very nice paper that has just recently been accepted in Bayesian analysis, I have to say. And this is a paper by Rajarsi Guainyoji and Dan Spencer on Bayesian tensor response regression apply in the context of activation studies. The important point here is that, as I mentioned before, we always have to be careful and probably we always have to somehow obtain some sort of a dimension reduction, right? Dimension reduction might be obtained, from star by managing our data, obtaining some summary statistics of a big data, right? Or by some more of some statistics, for example, by you looking at regions of interest, right? Or by looking at slices, like I just did in this example here, right? Another option might be just to implement methods of dimension reduction in our model, and this might be done, for example, by using tensor-based approaches. So these will soon be available on the Bayesian analysis website. Other questions? Are there questions? Sorry, no other questions. If not, at the end of the third hour, we get to the discussion of multi-subject modeling. As you have seen, only just for the analysis of a single subject, there are many problems that come to play and have to be considered, right? So think about then what is going to happen when we go from a single subject to a multi-subject case, right? So of course, it is even more challenging computationally to fit complex spatio-temporal models to voxel-wise data on multiple subjects. So the strategies are, for example, to group voxels into regions of interest and calculate summary statistics for each ROI. This is one of the typical way in which neuroscientists work in this area. And again, if you were to do a frequentist GLM analysis in this case, basically you will obtain the estimates of the GLM for single subject, right? And then you're going to use them somehow as representative of the as the starting point, let's say, for multi-subject analysis. So basically, by looking at instead of entire time series, condensing the information from single subjects, from each single subject, through the statistical parametric maps of each single subject, and working with them. In a Bayesian setup, we aim to be a bit more thorough in different ways. An easy way, however, to move forward is, for example, to the one that was proposed by Bauman in 2008, which is basically a two-stage model itself. So on one hand, first stage, one obtained the estimates of a voxel-wise regression coefficient in a GLM fit for each single subject. And the second stage basically combines all the estimates into regions of interest and basically build a hierarchical model where basically the new data, right, are given by the vectors, right, beta I, G, J, for each individual, each region, and each stimulus. So this is basically similar to, say, a Bayesian version of two or multi-stage models of fMRI, of frequentist statisticians, right? The advantage of this type of methods is definitely, computationally, you're going to summarize the spatial-temporal structure of the time series into basically these vectors of coefficients. So you're going to be able to conduct multi-subject inference quite normally through a simple hierarchical model, for example, on the second stage, right? By using basically the betas as the estimated beta in this case, right? Some estimates of the betas as your starting point, as your data. Computationally, certainly this helps a lot. And this is an example of posterior probability maps that he had obtained in his model. But I think that here this model allowed was also the estimate of the sigma square components, right, that are group specific, right? And through that, also he was able to obtain estimates basically of brain connectivity, but I'm not going to go much into the details of these. I think that somehow the literature has moved forward in that regard. Another paper that I would like to point out to your attention for multi-subject modeling is this paper by Nilotpal Sanyal and Marco Ferreira, and their approach is a bit different. So what they do is essentially to let me see if I remember correctly, right? Basically, this is their basic idea is to consider basically wavelet transforms, right? And obtain basically at the same time, but in a single stage setup, basically the wavelet basis as a combination basically of so basically the regressors, right? And I have to double check 1 second here one thing, but well, it's a bit complicated. I didn't want to go much into the details here, but the basic idea here is to use a wavelet-based transformation and have again some sort of mixture prior, spatial prior to describe with wavelet coefficients. I'm not, again, I wanted to point it out because it's a multi-subject model that has been proposed in the literature. It's not, and it is one of the first that actually tackled this problem in a single subject modeling framework from Bayesian perspective. But I might go a bit more into the self-promotion mode and discuss one approach that I have been working on with Marina Vannucci more or less recently, and previously in mine, but also something, another approach in that I will discuss following up to this, that has also been recently proposed and recently appeared in the literature. I am at 12 here, noon. I may ask, I mean, this is a good moment for me as well to take a break, probably again 10 minutes if you allow me. I'm getting a bit the sugar issues that comes with speaking for a long time. So if you have questions, please ask. Otherwise, I may see you for multi-subject modeling and then brain connectivity in the next hour. It's basically in 10 minutes, right? In 10 minutes for the next hour. Any questions? Okay. Are there any questions? But we check in you've probably seen a paper. I forgot. And I did not recall a few things. In particular, this is a multi-step approach. And basically, it's based on the estimates. I think right from what I got by writing the model for standardized regression coefficients, which are these, right? So I have to confess that I've seen this paper a long time ago, but I did not remember it as closely. I apologize for that. In particular, they use the variational-based approximation, so, which is important as usual for multi-subjects. Okay, that's live. I still. Okay, we are at 10 minutes after the last break. So, as you've noticed, the multi-subject framework comes with additional complexities due to the fact that we need to take into account the brain activity in multiple subjects. And so we will see these also later in discussing brain connectivity later this hour. One has to be somehow mindful of this aspect of the aspect of the computational aspects in this regard. And so in particular here, I'm going to describe a paper that I have been working on with a student of mine and Marina Vannucci a few years ago. And the idea here is we start again from the GLM framework. And basically, the idea is to extend the single subject regression model that we've discussed till not for the most part of this short course to multiple subjects. And in this regard, what we have is, so now, in addition to the bold response data at each times point, right, at each voxel, right, and these t times 1 vectors of responses. Now we have one of them for each subject. We indicate with I the subject indicators, and then we have basically the same GLM basically repeated for each subject. Now, in this context, again, the same problems as before, as outlined before for the single subject come into play, right? So how the beta vary spatially across a brain, but also at this point, what is the relationship between the activations for an individual and other individuals, right? So so are we able to describe heterogeneous patterns of activations across individuals? And notice in many cases and for many experiments, we may not expect that there may be, in terms of activation, very different patterns across subjects, right? If you're looking at a specific picture, let's say a picture of a beach, right? Many of us will probably have the same areas activated, right? But actually, an important interest in the neuroscience in this regard is actually understanding differential activations, even of same regions, right? How same regions may actually be differentially activated in different subjects. For example, one possibility might be, someone may react, someone that it is more empathic or less empathic may react differently, let's say, at the picture of a beach with respect to others, right? And this may actually be quite important in the study of behaviors. In a different context, you may think of reactions to, different types of stimuli, and see basically, for example, let's say if there is an interest to understanding the ability of a person to be following a particular treatment, let's say, that is interventional treatment, there may be some ability to understand or some possibility to use the information that we get by studying the activations, right, as possible biomarkers or possible predictive, in a predictive way, of the ability of a person to stay on treatment, right? So this is just to say that we certainly are also interested in describing and understanding the differential natures of the activations in different subjects. In this model, we consider the single subject regression model as before, but also as, in particular, we consider a long memory process for the errors. And if you remember the discussion that I had before, that basically entails, in order to get to a computationally feasible method, to describe, to move to the wavelet domain, use a Fourier transformation, and basically get the denoised errors, so where the errors are basically diagonal. Error matrix, variance covariance matrices are basically diagonal, depending on an innovation parameter psi and a delay parameter alpha. So, in a typical multi-subject study, one wants to understand the activation, but also, how different activation patterns may vary within and across subjects. One way in which one can think about this is by trying to cluster subjects that show similar activation patterns. At the same time, being able to capture spatial correlation among activations at possibly distant subjects. So on one hand, we would like to be able to characterize a cluster of subjects, and potentially, right, these cluster of subjects may be characterized by different patterns of activity, but not necessarily, right? Maybe just different levels of activities. But also, we would like to be able to capture basically and link together voxels, right, even in the same subject, that may be characterized by similar types of activities. There are a few ways in which one can do this, of course, but one possibility might be just to have a sort of prior that induces basically clustering across individuals, but at the same time sparsity, right, across the within of the patterns within each individual. So what we proposed in this case was simply a multi-subject spike and slab prior. Again, here the idea is that there is a indicator, a latent indicator gamma, that identifies if a voxel is active or not. If gamma I nu for this individual, voxel V is not active when gamma I nu is equal to 0 and beta I nu is equal also set to 0. Instead, if there is an activation of the voxel at this voxel for this individual, beta is taken from a distribution GI. And this distribution GI is a distribution of activations which is subject specific. We want to relate together the different subjects. So a way to do this is by relating together in some ways the distribution. And the way in which we did this was basically by using what is called a hierarchical Dirichlet process structure. This induces basically sparsity within subject, but also relates the distribution of activations between subjects. I'm not going to go into the details of the technique. Again, tomorrow, Cottas will be able to go more in the details of Bayesian parametric priors. Here, the basic idea was to use a hierarchical Dirichlet process to be able to describe the activations within subjects on one hand and enforce somehow or encourage better sparsity. And on the other hand, try to relate the activations across individuals. In order also to allow the possibility to take into account within each individual that activations at nearby locations may actually be similar, we also used a spatial variable selection prior, right, based on the Markov field prior, which is what I had discussed before. So basically the idea here is, again, that the probability of activation in each individual at each voxel is going to depend on the activations for that individual in voxels nearby. For posterior inference, one can follow a Markov chain Monte Carlo approach, but if a Monte Carlo Markov chain MCMC approach is followed, there are of course some limitations in the type of data that you're going to be able to use. In particular, for example, in the number of regions that you may be able to consider. So a possible alternative is instead to use a variational-based approach. And you will see that when going to the multi-subject studies, unless you really have a small number of subjects and you've been able to do some sort of a dimension reduction beforehand of the data, it's going to be very difficult to be able to apply full MCMC. Here I had a presentation of the differences between MCMC and variational base. In this case, I'm going to skip most of it. I'm just going to say that, of course, the MCMC is going to provide good performance on activation detection, good estimation results for the model parameters. However, from a computational perspective, it's going to be very expensive. However, the variational base is going to be more computationally efficient. Of course, you're going to lose something. And in particular, here, you're going to lose something in terms of false positive rate. Notice that major drawbacks of variational base is the fact that the variance of the betas, right, is somehow underestimated. In that regard, we are not really interested much in the variance of the betas, right? But we're more interested, right, here in the activation detection. So in terms of activation detection, let's say that it does reasonably well. However, of course, it comes with a slightly higher false positive rate. We can look at some results quickly. Simply, these are data on 27 subjects. And in particular, we look at two particular slices here. So we are also doing some dimension reduction at this regard, right? And the goal was to detect brain activity in response to visual scenes. And we compare our method again with the state of the art, which is SPM8. And this is for different subjects. And SPM8 actually does single subject, right, analysis, in the model that we had implemented. So on one hand, one can see the advantage of Born strength across subjects, but also, again, some advantage coming from the fact that we are using priors that induce both clustering within subject and across subject. So the priors overall take into account also, in particular, the two subjects the two clusters that we identify are not really distinguished in terms of brain activities, right, of regions, but mostly also in terms of intensity of brain activity in those two regions, which is something, again, to be considered the heterogeneity across individuals. So the priors take into account the structural feature of the data. They take into account the structure of the spatial information that we have, or the fact that the data are temporally correlated, or trying to address the heterogeneity of individuals. Eventually is going to improve somehow the performance of activation detection. Again, for multi-subject methods, typically one has to go somehow to consider Bayesian approximation methods alternative to full MCMC, and somehow work with those. I wanted just to give a shout out to the work of a former student at Rice, who has actually developed for the single subject and multi-subject method that I have discussed earlier in more in details, in particular this last one, the multi-subject method, a tool that a MATLAB GUI that basically implements the two methods. And you can see you can go on the GitHub of Eric, the student, and basically download the tool, which is a Windows-based tool for the analysis of fMRI data. So this is a good point to stop, also because we are at the end of the methods for activation detection. I went a bit longer on this than I thought, but I thought it was useful. So I actually had a question before I see that one question was, apart the use of low rank modeling, tensor modeling, are there any other applications where the authors use dimensional reduction and or linear or linear projection techniques prior or during the modeling step? Yes, there are many. And as I mentioned, actually, it is important in this type of studies, especially for multi-subject, to consider typically dimension reduction techniques. One way in which this is done, for example, is also to use some PCA, or better in the context of neuroimaging data, as typically independent component analysis is used. So basically, the idea is to reduce the dimension of the data to a manageable number of factors, right? Or of time series, right? That one could use. Maybe another possibility, similar to what you do, right? Basically, by considering, instead of entire whole brain analysis, by considering sets of regions of interest. I don't know if that helps or answers the question. We will elaborate more, and we will see more examples as we go of sort of ways to simplify the complex problem. The use of independent components, for example, is actually well accepted in the neuroimaging community by neuroscientists. Another question, can you please elaborate a bit more on how the HDP record data process induced clustering among subjects in this last work? Has anyone considered applying the nested DP or its modification to this type of data to cluster both subjects and activated voxels? So for the second question, as far as I understand, nested data process, which is a model where basically instead of, I didn't go into the details here, so this is my fault. And again, the data process basically clusters the GI here as so the nested data process basically consider the GI as a whole distribution. The architecture process instead consider the GI as distribution characterized by some atoms that comes from a bag of atoms, let's say, right? It's a slightly different ways to see the clustering of the betas. So as far as the second question, nested data process, no, I haven't seen any. I haven't personally seen any case where the nested data process has been used. As far as the first question, can you please elaborate a bit more on how the HDP induced clustering, among subjects? In this case, the clustering was basically given by the clustering of the beta I, right? Basically by the clustering of the effects, right? So it's, if you want, it's indeed a bit more of an indirect way, let's say, to show basically that it was more related to the betas, right? We had to do some sort of post-processing, right, of the betas using basically known ways to get from the output of the data process model, right, architecture process model, but to get basically to this cluster, we had to do some sort of post-processing eventually to define this cluster. So it was not as direct as I showed it here. Yeah, it's basically based on techniques that have been used before as well in the literature. Okay, good, great questions. Okay, so now let's move on to the second part of this short course, and in particular, in the next hour or so, I will discuss, will try to discuss the topic of brain connectivity, which is indeed a big topic today in the analysis of fMRI data. So connectivity looks at how brain regions interact with each other and how basically information is transmitted among them. Let me make sure that there are no questions. So the idea is to try to understand how connectivity may also change in different subgroups of subjects, or maybe also sometimes in the same subject, right, in different moments of experiment. And we will discuss that in detail. So one may look at both task data, when it is data where a picture, for example, or different tasks are asked at the individual, or instead a resting state data, basically seeing how the connections of the brain while the person, the individual, is resting in the scanner without being asked anything, to do any task. So why one may want to study connectivity? Well, mental health disorders and brain diseases, mental health disorders can be described often as disruptive the connectivity patterns of individuals, right? As a disruption of connectivity patterns in individuals. Similarly, some brain diseases may induce somehow some connectivity changes in individuals. So the study of brain connectivity in this regard is seen as a possible, not only statistically interesting, but also clinically interesting type of goals. So if you look at brain connectivity, however, we have to be aware that in the study of fMRI data, in particular, two types of connectivity have typically been considered. One is what goes under the name of functional connectivity, that is the undirected association or temporal correlation between Bohr signals from remote, especially remote brain regions. The idea here is to think basically at correlation, right? Somehow between the time series at different voxels. The other notion is the notion of effective connectivity, which describes the direct influence of one brain region on other regions. So again, I mentioned this at the beginning of this course. Functional connectivity relates more to correlation. Effective connectivity tries to infer in some ways causation, right? And I'm using double or triple quotes here. So in terms of functional connectivity from a Bayesian setup, there is quite to be some work in the literature. Typically, both in the Bayesian and frequency setup, typically in the frequency setup, one looks at correlations between regions of interest, or instead between a seed region and other regions of the brain. One looks, tries to obtain and understand connectivity by somehow working with independent component analysis or other multivariate methods for dimension reduction. Another way in which connectivity has been studied in frequency setup has been often by the use of graphical lasso. In the Bayesian setup, instead, the models for brain connectivity have been a bit sparse over time, but they are becoming more and more prevalent these days. So in particular, here I'm going to just mention briefly a paper by Patel and Bowman in 2006. Basically, their goal was their model basically was to first threshold the time series to detect activity, and then basically based on the threshold time series, and basically creating sequences, binary sequences of 0, 1, basically compare the expected joint and marginal probabilities of elevated neural activity between regions. This second paper actually is a paper that we have already described earlier, the two-stage model by Bowman and others. Basically, they allowed in their parameterization, there was the possibility to take into account both short range and long range connectivity between regions by basically working with the variances of a model. And then in the single subject case, the paper that I described before, actually, if you remember, was clustering spatially remote voxels with based on the innovation parameter and the delay parameter psi and alpha, right? And basically there was clustering across space. So all these methods were methods that allowed in different ways to describe somehow patterns of correlations across different voxels. I'm not going to go into the details here because I think that it is more important to discuss more recent developments in the topic of brain connectivity, and in particular the fact that you know, all these methods that I've been discussing, right? They basically took the time series and they got at the end a sort of correlation pattern, right? Across the entire experiment, right? The idea basically to study, the underlying idea here was to study the patterns of connectivity, the correlation between time series along an entire experiment. Now, it's clear that my connectivity networks, right, may change during an experiment, right? There is an increase in realization, right, that although traditional approaches rely on the assumption of spatial and temporal stationarity throughout the measurement period, in reality, brain connectivity is by nature dynamic and fluctuating, right? Both across tasks and in resting state. At the beginning of this short course, I was more active and alerted, right? Now I'm kind of feeling the time going, right? And so similarly, in an experiment, fMRI, although 5 minutes long, one can think that, for example, people may experience different states of connectivity during the experiment, right? If I'm the scanner, I start thinking about something and then I start thinking something else, right? If I'm in resting state, right? So I'm not going to be exactly, even if I'm in rest, I'm not going to be exactly in the same state over time. And similarly, during a task-based fMRI, one may expect that the connectivity patterns may change depending on the task. So there is this realization that there is in reality dynamicity, right, and fluctuation of connectivity patterns over the course of an experiment. Of course, if we realize that, there are some statistical issues that comes clearly into mind. If you're thinking that there are a sequence of states, right, appearing or characterizing our connectivity over time, we of course have to consider the fact that we will have to somehow describe through our statistical model the persistence of a connectivity state over time. We will have to identify change points, right, between different network connectivity pattern, right? Once we have obtained the change points, we also have to identify to estimate the networks between the change points. And we have also to assess the differences across the networks. All of this that I have described is simply for a single subject. What happens and what are we going to do for multiple subjects? So, so again, the type of challenges, luckily for us as a statistician, compound, right? And that makes the topic quite interesting. So I'm going to describe two recent approaches to the study of what goes under the name of dynamic brain connectivity, dynamic functional brain connectivity. And the first one is a recent paper by a student of mine, Ryan Warnick, and together with Marina Vannucci and Vince Calhoun, who is now in Georgia. And basically, a Bayesian approach, we are going to present a Bayesian approach for estimating dynamic functional network connectivity in fMRI data. Before I get into the model specifications, I just want to let you know what frequencies do in these cases. A typical model in which this is taken into account by neuroscientists, right? So we have a sequence of time series, right, for each voxel, right? And basically what we do is to use a sliding window approach. There is basically a sliding, we define a sliding window of arbitrary length, right? And in each window, they basically estimate a windowed covariance matrix, basically based on the window that they have decided. This can be done in different ways, right? It could be simply a simple, based on simple variance covariance matrix, or it will be something a bit more, let's say, more sophisticated, like for example, using GLasso, right? Once we have basically at the end, when basically we have for each one of the windows, we have a estimated variance covariance matrix. And then basically what we do typically, the typical pipeline is just to cluster the matrices through some simple clustering method, for example, simple k-means. Now, as statisticians, we clearly recognize the issues with this approach, right? Just without even going much further, just the starting point, the definition of a sliding window, right? What's the length, right? And just this alone is a point of contention. And indeed, a good statisticians, Martin Lindquist, in a paper in Neuroimage, mentioned this issue and first showed that, well, with a simulation that was made of pure noise, right? By choosing different sliding windows of different length, one could actually show something, even if there was nothing going on. And then Martin Lindquist basically mentioned that in order to try to analyze this data and this problem of dynamic connectivity, instead of relying on sliding window methods, we should probably consider time series models or models that, state space models, right, that are typical in the time series literature. So try to use that information. I'm not going to go over the other issues that one may find, right, in the use of the sliding window approach, but one of them is, of course, again, if one uses, for example, a GLasso approach, right, for sliding window, and then moves forward from that for further estimation on the inference, of course, also the problem of selective inference comes forward, right? So in a Bayesian setup, how we could go about defining a model for dynamic connectivity. Well, let's go back to our simple model initially, right? And just consider, again, fMRI measurements on V neuronal units over T time points. These neuronal units can be regions of interest, or can be also time courses obtained by a PCA or independent component analysis done before the use of a model. So we have these for different subjects. Here I'm putting multiple subjects, but indeed what I'm going to consider right now is a single subject model. So I apologize for that. This is not really a multiple subject model, the one that I'm presenting. But so the model is going to be characterized by an error component and a regression component, right? And as before, right, we can put ourselves in the general linear model, right, where we have maybe some possible covariates if you're looking at different subjects, right? But again, this is not the case that I'm going to describe today. And then we have instead our general linear model XT beta here, which basically has the convolved stimulus from the fMRI experiment. If, for example, we are considering a task-based data in this case. Anyhow, the main point here is, for a second, let's forget about the characterization of the mu t. We're going to use the epsilon t, the variance covariance structure for the errors, to define our connectivity model. And in particular, so here epsilon t now is a vector, right, of v-dimensional vector, right, over time. And we want to describe basically the connectivity across individuals. So the type of model that I'm going to describe is what goes under the name in the literature of a task residual connectivity model. But basically, I'm going to describe the residual connectivity after I have considered, if I'm in a task-based case, in case the individual is actually responding to a task, the connectivity patterns is basically just going to be captured by the characterization of the error model, right, of the residuals. And in particular, what I'm going to consider is a Gaussian graphical model on the errors. A Gaussian graphical model is a convenient model to describe connectivity, because the presence of connections, right, let's say edges between different voxels, is completely explained by the precision matrix. In particular, so if there is a 0 in the precision matrix, right, that means that there is no edge, there is no connection between two voxels, right? So at a first level, so I'm going to assume a Gaussian graphical model on the errors. This will allow me to describe the connections through the elements of a precision matrix. Any elements that is non-zero of a precision matrix is going to describe a connection that is present between two voxels. Another point that I will add, however, is that the connectivity pattern described with precision matrix is not going to be the same over time. This omega here is going to depend on a variable st. That this st is a state variable that changes over time. So basically, my Gaussian graphical model in this regard describes connectivity as a task-based, task residual method by describing the precision matrices as of the residuals as changing over time. And so ST then indicates the connectivity state at time t. What we want is basically to describe different connectivity states at different times. But of course, we also have an expectation that the connectivity states may not change abruptly from one time to the next, right? So we may have an expectation that there may be contiguity of states over time. And we may also have an expectation that there may be a finite number of states that we may encounter during an experiment. So all of this just to say that a simple model to induce change over time in the omegas is simply to consider a hidden Markov model. Hidden Markov model to classify the mental states. In this case, so there is this latent hidden Markov model will allow us to understand, right, or to capture change points in the connectivity through the change points, through the change of state, right, as dictated by the latent hidden Markov model. So the observations that we obtain from our fMRI data will depend on latent neurological processes that are indexed by ST, where ST is driven by an hidden Markov model formulation. Now, the patterns of that are characteristic of each state are captured by this omega ST. There are two things to consider here. First, what we're going to consider initially, a very simple way to do this is to consider a finite number of states, right? The number of states must be decided before the analysis. Of course, there must be some a priori information that may drive it. alternative possibility is to choose the number of states based on some model selection criteria, right? Maybe some DAC or WAC or so on, right? That's a possibility. In some cases, like, I mean, since we're considering here task-based experiment, at the minimum, we're going to expect there may be different types of connectivities for different tasks, right? So that's the minimum assumption. But of course, one may assume that there may be more states than the tasks themselves. And the second point is we need to have a model, right, that describes the connectivity states dependent on that are active at each the connectivity patterns that are active at each state, right? So a model for the omega ST. And these can be done again in different ways. One way that we followed here was to use basically a graphical Wisher prior on the omega ST, where basically the patterns of connectivity are going to be dictated by an underlying graph that is active at each time point, so GST. In particular, this underlying graph is going to dictate the edges, right, between the different voxels, different regions. It's going to dictate the connections between the different voxels, and the graphical Wisher prior will basically be used to determine the magnitude, right, of the errors of a precision better in those points, the magnitude of the correlations, if you want, if you think about partial correlations derived by the precision. Anyhow, so this is just to say that the omega ST is going to be based on an underlying graph, and indeed the different patterns of correlation, the different of connections will be described by the underlying graph. So in order to describe the different patterns, what we're going to use is the idea of having a supergraph. Here, the idea is that we do not expect in general that the connectivity of one state be completely independent of the connectivity at another state, right? So we may expect that there may be some sharing, right, between the different graphs. So if there is a connection in a graph, so if there is an edge between two voxels in one graph, we may expect that other voxels, so the same connection may be present also in other graphs, or maybe not, right? So in order to do that, we use a Mark random field prior linking the graphs. This is an idea basically of having a super graph, and in particular, we're going to increase the probability of the edge presence in every graph by the consideration of basically relatedness between the networks. So if we think that two networks are somehow related, we're going to have a parameter here that's going to increase the probability that if an edge is present in this network, it should be present also on this network. The estimation of these parameters is going to be all obtained through our Bayesian model. So this allows us flexibility because we can share information across networks when it is appropriate. Also, we are also being able to share information across related networks as well as across subjects if we are going to apply these extending it in multi-subject studies. And somehow this idea of a supergraph is related to the idea of multi-layer networks, right? Which has been gaining traction in the neuroimaging literature recently. I'm not going to go much more into the details here, and I just want to show a bit some analysis, right? So this is an application to a fMRI study with basically where individuals were left in the scan. At some point, they had there was like a beep, right, an auditory stimulus, and during the stimulus blocks, the subjects keeping the eyes closed for the duration of the scan was presented with the binaural audio tones of varying frequencies at different intervals, and they had to press a button in response to each of the stimuli as quickly as possible. And what we see here, we just consider actually we obtained after model selection criteria through DSC that actually two states seem to be good enough to represent this data. One state represented somehow the resting times, and the other state representing the stimulus present. But you can see that overall, so this is the posterior state probability of state 1, and possibility of state 2 in this case. Overall, the posterior probability of each stimulus changes over time, and they somehow overlap with the presence of a stimulus. So when the stimulus is present, in particular, you can see that the probability of what we call here state 1 increases, whereas the posterior probability of state 2 increases with respect to time intervals that correspond to the absence of stimulus. I'm not going to go much into the details. I just want to keep going 1 second, and this will be our last point before the next break, which will project us into the analysis of EEG data. But there is also another nice paper that I'd like you to have a look at. This is a very recent paper by Lukemeier, Kundu, Pagnoni, and Guo, which is being recently published in JASA. Actually, probably this is still, I have to check if it is still, yeah, it is still on, I got it this past week, so this is still in advanced publication. And they also propose a Bayesian joint modeling on multiple brain functional networks. So in this case, they are actually looking at also, I think, at we're also looking, sorry, we're looking at multiple subjects, and they're looking at different experimental conditions, and they're basically interested in studying basically how different networks characterized in different experimental conditions change or can be estimated during the course of experiment. So the goal is to estimate multiple networks using Gaussian graphical models again, characterized by sparse precision matrices. Let me see, I didn't check if there are questions, but of course, please feel free to ask any question. So again, so this is a very recent paper, and the idea here is to consider, so in this case, they use the pre-Whiten fMRI measurements for each experimental condition. These, and basically their interest is basically just in estimating the precision metrics, right, that corresponds to each condition. And in order to do that, what they use is simply basically a spike and slab lasso approach, right? So if you followed the discussion by Linero and Jose Antonelli yesterday, this should be clear. But the basic idea here is we're going to have on the elements of the diagonal, so on the diagonal elements of the precision matrix, they are going to have basically an exponential distribution. in the off-diagonal elements of the precision matrix, which are the elements that capture the presence, right, or the strength of the connection, right, we're going to have a spike and slab lasso. And so it's linkage prior. So as so in this way, we are going to obtain and obtain sparsity, right, of the connectivity patterns, right, by using a spike and slab formulation. The other point that they have is that the weights of the spike and slab prior are going to be given by this WGKL, right? This WGKL actually represents the probability of having a functional connection or not, right? And in order to do that, to model this WKL, they follow a somehow, complicated but effective model. Basically, they use a parametric link function that considers a general let's say, baseline common shared effect and differential effects for each group. The idea here is that there is an in response to the task, there may be a general overlay, I mean, there may be a general base probability, again, of activations. Think about it's somehow similar to the ideas that you've seen before for the Markov field prior, right? There is a general probability of connections between two regions, but then there is a hump, let's say, more that is given by the a group specific, right, or task specific effect, right? And we do this by, we also induce clustering between the task effect, right, and within the task effect, right, they and basically create again by inducing this clustering, they encourage again the possibility of first, they're going to reduce the dimension of the parameters in dramatic way, but also they sort of regularize somehow the problem by getting clusters, right, of the parameters across the different connections, right? So. Okay, so yeah, I could have probably just gone to this slide under the specification, but baseline effect estimates the shear, the probability of a connection by pulling information across experimental condition. Instead, the differential effects contribute to the network-specific variation, right? And one can also identify, right, and try to understand by analyzing the values, assume the parameters etas, right, the differential networks and the probability of a differential network that are estimated. So this is a very nice paper that has been this has been recently proposed, published in the in JASA. Okay, we are almost at the end of our 4th hour, and what I'm gonna miss to discuss is basically models for effective connectivity, which somehow try to get at the causal dependencies as opposed to simple associations with respect to connectivity estimation. And the models that are typically considered are different, right? Typically, one may consider to discussing describing effective connectivity, structural equation models, dynamic causal models. For example, Freestone has worked a lot in this area. Then Bayesian networks. A promising area of research is given by vector autoregressive models in particular, and in particular here from the notion of Granger causality. And where the idea is basically that the causality is captured by the fact that the effect may precede, may come after the cause. So the cause precedes the effect. So there is a temporal there is an expectation that the cause of an effect, what happens in one region, may affect what happens in a region afterward, right, in a subsequent time point. But I don't have much time to go through this, and I think I have to make, unfortunately, the decision to sort of jump through this. I'll do this for two reasons. First, because I think, I mean, we are kind of being short of time right now. But also, the second reason is because vector autoregressive models are somehow studied extensively, and we will go back to them actually a bit in the next slot, which is going to cover the use of Bayesian methods for EEG data. So I will take again 10 minutes break, and if you have any questions, please feel free to ask. And of course, if you have also any suggestion, Oops. Again, if you have any questions, please feel free to ask. Yes, we're all tired. So where are you guys from? I can recognize some names. What time is in Italy now? France? Ten, 12 in Italy, okay. Thanks for being here. Mexico, great. Thanks. Mexico can be the same time as ours, right? As I am or not, right? But thanks. This is a nice thing about the ISBA meeting this year, that allows people to connect from different areas at very different times. And it is a huge program. 2 p.m. Yes, Mexico City. Mexico City 2 p.m. is 1 p.m. 1:10 here. So what time did I stop? Do not remember. So let me know if it is okay, if the pace is still good. If there is anything that you'd like to discuss. Some questions that you might have and would like to see maybe addressed, or, you know, general questions, things that we have not seen. This is a good question. Have you considered the interpretability of the models? Yeah, I mean, typically, the good way to work with this type of data, right, with type of fMRI data, if typically if there is a collaborator with you. Typically the collaborator is interested, right, in the interpretation of a model. So being able, so I didn't expand much on that, but for example, the idea that I was making, oh, sorry, but I was making at some point, oops, here, right, but there were two different clusters. These sort of clusters, for example, have been discussed with the collaborator, and in particular, the idea that there may be different types of levels of activity, not only just pattern, right, is was indeed nested also in the idea that on the type of expectations that the collaborator in this case was looking at, trying somehow to identify clusters of individuals that were characterized by different intensities because these meant something for them. In particular, the idea that one may be more taken by some reward sensitivity, maybe one individual may have higher reward sensitivity than others. Yes, uncertainty quantification on data and model, that's another important point. It's definitely important. So once you have, of course, given a model, you want, certainly, I mean, this is, of course, just a plot of the posterior means in this case, right? But you want to make sure, right, that what you get is, so the type of inference that you have. At some point, I described a bit sort of different types of, you know, of the variability, right, for, of the estimates, right, for probably this is not the correct picture, but anyhow, so it's something that you have to do. Somehow I swiped all this discussion, all these points, very important point, under the rug, but definitely, I mean, it's important in terms of also model selection, right? So in this regard, often simulations, in particular, that simulation that try to mimic the fMRI data the most instead of being generated by our model, right? So there are some tools out there in the community, for example, that allow you to design an experiment and basically generate fMRI-like measurements that these tools are typically developed by neuroscientists, right? And so it's also good to try to test your models on this type of fMRI-like data. So I don't know if this actually answers your question, but maybe we can discuss further. If you want, let me know if you want to discuss more on that. I'm talking about Alessandro here, and in case I can also give you the possibility to chat. Oh, okay. I'll have to think about the literature on uncertainty quantification. Right. The other big trend that I see in the questions, right, is computational time for the models, and if there is, for example, the need of a programmer for this type of models. Well, at some point, all statisticians are becoming a bit some programmers at some level, but indeed, the computational cost is high often, right? Depends a bit, but there are ways, as I mentioned, that can be used. There are possibilities to try to reduce the computational effort. One way, so for example, so there's also, so in particular, when moving from single subject to multiple subject. So overall, I mean, so for example, for these type of models that I presented here, right? I mean, overall, it is not for a single subject. I mean, I don't remember in particular for this model, but I mean, but I did. It was probably, you know, not taking long, right? I think like half an hour, maybe to get the answer from what I remember, right? It was some time ago. And as you can see from the different types of years that I put here. So definitely, I mean, but this was a full MCMC implementation. If you consider, for example, this MP-based fMRI, MATLAB GUI, in this case, if you can use it, you can try by yourself. It's based on a variational-based approximation, and it is quite fast in the matter of minutes, if I remember correctly. So it depends a lot also on the tools that you're using to try to speed up computations and the use of approximate-based computations, approximate-based approximations, approximate-based solutions, or Bayesian, well, variational base or other approaches can help. Does that answer the question? Good. This was great. These were great questions. So thanks. So let's start to discuss a bit, and again, probably I won't be able to cover all I wanted to do, but there is quite a bit of work also for analyzing EEG data currently in the literature. But before I get to the most recent work, let me first remind you a bit of what we're talking about here. I described this at the beginning of this short course. So some characteristics may have been lost by now, but basically what we're looking here are data that are obtained by measuring the electrical activity of the brain from the scalp. There are some electrodes that are basically put close to the brain, right? This is a non-invasive procedure. Typically, you can think about 128 electrodes, right, on the brain, and basically, again, the individual may be asked to perform a task, and for each one of the electrodes, one gets time series that are typically characterized by a high temporal resolution. So these time series reflect the cortical electrical activity. So these are typically time series, right? And although sometimes these time series are summarized by neuroscientists in sort of a functional data analysis forms, but I'm not going to discuss about that. The way in which we are going to discuss them today is through as time series, and in particular, the analysis of this data typically follows two different directions. One is the analysis of the data in the time domain, and the second is the analysis of the data in the frequency domain. But if we want to talk about Bayesian analysis of EEG time series, right? And as I said, there is a renewed interest, as I've seen in the Bayesian literature in the recent years. But if we want to talk about this type of analysis, I think we cannot but discuss the first papers. And the first papers are actually papers that have somehow created sort of initiated sort of discussion of analysis of Bayesian models for EEG data, and they are due mostly to Mike West and Raquel Prado. In particular, here I am going to discuss. So there are two major papers. One paper in 1999 by West, Prado, and Cristo, and basically they consider basically methods for decomposition of basically they basically decompose the individual time series basically into collections of latent components at different frequency bands. Here today, I'm going to discuss a bit more the second paper, 2001, by Prado, Mike West, and Crystal. And again, I'm going to give just simple highlights of the paper themselves. So they consider models for non-stationary time series for multiple EEG series generated from individual subjects in a clinical neuropsychiatric setting. So this model was still a model for single subject analysis. And in particular, they discussed the use of dynamic latent factor models and dynamic regression models for the analysis of EEG data. So let me just give a bit of a presentation of the type of models that they consider. In particular, they consider, in this case, a simple set of EEG data. They have 19 EEG channels recorded in 8 subjects in multiple conditions. However, they do a single subject analysis for each one of them. And basically, one of the conditions includes a electroconvulsive therapy seizure, ECT. which is the interest is indeed studying the ECT seizure. So in order to do that, what they consider is basically a dynamic factor model. And suppose that you have M time series that correspond to the M subjects, and they have the observation recorded at time t on each electrode. M time series correspond to the M electrodes, to the M electrodes, right? And suppose that we have recorded at each time t on each electrode, we have M electrodes, sorry, not what I was thinking. So the basic idea here is to have our time series YIT related to a latent process XT. And the XT process is simply a time-varying autoregressive model of order P. Time-varying autoregressive model basically means that XT is related to previous value of the same latent process at the previous lags, right? And in particular, the time-varying aspect comes into the coefficients, right? So basically, the coefficients for each lag phi tj follows a simple AR1 process, right? So again, yi depends, so the time series at each time point t in each electrode is going to depend on a latent process xt, which follows a autoregressive process of order p, but also we assume that the type of that the coefficients of the autoregressive model are time varying. This is essentially a dynamic linear model for those of you versed into time series. And since it essentially depends, the time series essentially depend on latent process xt, this is essentially also a representation of a dynamic factor model. And an important point that they consider is the possibility that the observations YIT depend also on the lag or lead that the observation time t exhibit with respect to the latent process. The idea here is simply that the signal at one channel may be delayed with respect to the signal recorded at another channel, differently between different moments of the recording. So you want somehow to capture this type of different leads. So the idea is, for example, that during a particular, during the seizure, one may see, for example, a different type of or maybe more a faster, right, relationship between XTs, right, between the latent process and the Ys, right? So, and the observations. So, in other words, we want to explore models that explicitly involve time-varying description of the amplitude ratios and the lag lead structures across the channel, with the idea that basically there may be different types of interactions between the latent processes, right, at different moments of the experiment. So this was a univariate time series model at this level, right? This was a 2001 paper. So they actually did this analysis for different time points, right? And one thing that one may notice is that you know, the posterior means of the dynamic factor weights for all for the channels, right, during at the beginning, at the mid, and toward the end of seizure show some changes, right? In particular here, one can show that during the mid of a seizure and the end of a seizure, they commented there was some sort of asymmetry that had to be somehow, that was interesting to study, right? But also had to be more understood. So again, so this was a first attempt and one that has been impactful in the literature in terms of reference, right, for studying EEG data using Bayesian methods and sophisticated Bayesian methods in this case. So, and there have been some other papers that I would like to recommend to your attention. For example, this paper by Raquel Prado, Francesco Molina, and Gabriel Huerta, where basically they use and extend the approach to vector autoregressive modeling. In particular, they consider a mixture of experts. So basically, the idea here is that there is indeed a mixture distribution that describes, if I remember correctly, the latent structures. So, this allows basically to describe basically overlapping structures, basically. So anyhow, so time domain models are typically based on vector autoregressive models in this regard. The other big, other important important way to describe models for EEG data is through spectral analysis. And I don't know how much familiar you are with spectral analysis method, but basically the basic idea here is basically the time series is decomposed into a set of waves that oscillates at different frequencies. And the primary tool for spectral analysis is the spectral density function, which is also known as the power spectrum. So under mild assumption, the spectral density uniquely determines the properties of a stationary time series. So we can basically represent the time series through its spectral representation, and by the inspection of the power spectrum, we can determine the properties, or we can analyze the properties of a stationary time series. For example, if the power spectrum is of this type, we can see that the model is characterized by high frequency data. So time series fast oscillating. Instead, if the spectrum is of this type, we can see that there is a bit low frequency oscillations, right? So there is going to be a more smooth time series over time. So together with the power spectrum, so the spatial spectrum represent basically the proportion of the total variance of a time series that is explained by the waveforms that oscillate at each frequency. And so there are some methods that have been employed to estimate spectral density. Typically, the many methods rely basically on parametric assumption, but overall, we can also obtain a non-parametric periodogram. But overall, in order to obtain a based, so this is the non-parametric periodogram of the that is an estimate of the power density. But then in order to estimate the power spectrum, we may need to make some sort of parametric modeling here. Typically, what we need in order to obtain, so in reality, to obtain good estimates, right, is somehow to enforce some form of smoothing. So here in this example, I'm assuming, so starting from the periodogram, which is a non-parametric estimate of the spectral density, and this being the truth, right, by simply smoothing the periodogram in some ways, using, for example, a kernel representation, I can see that I can get closer and closer to the truth as I increase basically the level of smoothing. So in other words, so the takeaway message from this quick introduction to spectral analysis theory is the properties of a time series can be represented by the power spectrum. And in particular, if you look at the analysis of the, we look at the spectral density, and we try, and we want to do a spectral analysis, what we need to do in order to get to conduct a likelihood-based inference, right, is to reframe the problem, right, using, if you want, so basically coming up with a good likelihood model for this type of data. So a typical way to frame the likelihood, the modeling framework with spectral data is by using what goes under the name of Whittle likelihood. So when conducting a spectral analysis, the Whittle likelihood is often used as an approximation to the true likelihood. This is basically a likelihood function that depends on the spectral density and the discrete Fourier transform of a series. And in particular, this guy here, right, is the periodogram, which is the sample-based characterization of a non-parametric estimate of a spectral density that I have described and I was considering here. So based on the sample periodogram, basically, we can obtain a likelihood function, and this is basically the idea behind the use of a Whittle likelihood. So the use of a Whittle likelihood is one way in which spectral density spectral analysis is conducted, right? And at this point, we will have to basically build a model based on this object, trying to describe the spectral density in some way. Okay, so a few things. So this is just to give an example, a quick introduction. I know that I've been very quick, very succinct, very basic, right? But I hope that the basic idea have come forward. And the basic idea being in this case that we can do in some ways likelihood-based analysis also when working in the spectral domain. And typically how this is done is by using this instrument, which is given by the Whittle likelihood. However, recent methods actually have considered slightly different approaches. And I want just to describe them a bit here. And this basically brings us from 2001 to 2019 in a jump, right? But again, so as I mentioned, Raquel Prado has been one of the major players in this area from the Bayesian perspective. So there is a nice recent paper in JASA that discusses Bayesian spectral modeling from multiple time series. And the basic idea here is simply to, instead of considering the Whittle likelihood, basically to replace the Whittle likelihood with a different type of construction. And in particular, their model assumes that there is a mixture distribution, a mixture of Gaussian distributions that characterizes the distribution of the data. And the mixture distribution has weights, G, K, W, that they define in a sort of adaptive way, and I'm going to discuss this in a second. So, these parameters, so an important point to notice, right? These are parameters that depend on the frequencies, right? So we are basically describing here the information from this periodogram, basically, right? Okay, so if we do this, basically, so we basically substitute to the Whittle likelihood this mixture of Gaussian distributions. And again, important point here is this weight, and this weight here is actually modeled by a sort of flexible, in a flexible way, as basically the difference of two of consecutive distribution function on 0, 1 between the points k minus 1 over k and k over k for different k's, right? So it's basically the idea here is to use a integral transformation to define the weights. So this allows basically, so since this construction is the same for each omega, for each frequency, there are basically a different set of weights which evolve smoothly with the frequency. And they also provide an extension to this model, which is basically just for a single spectral density to multiple spectral density, but I omit this description. This is just to say that although the Whittle likelihood is used most often in the analysis of spectral data, at the same time, there are ways to overcome that, and in particular, Cadonna, Cottas, and Prado propose a flexible mixture distribution, which actually comes with all the advantages of working with mixtures instead of more complex objects. What time is it? I see that my time is always shrinking. I wanted to point out also to other different models that sort of improve, right, on the simple Whittle likelihood approach that we've seen, right? And in particular, there are these models by Rosen, Wood, and Stoffer, and in particular by Rosen and Crafty. Later on, we will see a couple of examples. And basically, their idea is the following: instead of analyzing the time series as a whole, let's just let's just divide the time series into a finite number of segments and estimate the local spectra in each one of the windows, let's say, by smoothing spline. And basically, this basically allows us to use the Whittle likelihood for each one of the segments, but the likelihood function for the entire time series will be approximated then via a product of local Whittle likelihoods, right? So it's again an improvement of the simple typical approach that it is used in spectral analysis. And again, fast forward to more recent time, and there are a series of papers by Robert Crafty at University of Pittsburgh that also work on with Bayesian methods, right? And they work with partitioning a time series into a known number of approximately stationary segments, and allowing the components, with different spectral components to change in the different segments. And the particular model that we propose here is what we call an adaptive model, but in particular allows to approximate both abrupt and slow. And finally, even more recent, 2021, as you can see, they extend their framework again on the analysis of the spectral analysis method to high-dimensional non-stationary time series. In this case, we're going to consider, as I mentioned, some sort of dimension reduction technique. In this case, tens product of penalized splines and multiplicative gamma process linkage priors to describe or to tackle the high-dimensional problems in the analysis of EEG data. So this, again, as I done before, I sometimes I wanted to give you a few examples of things that people have been doing. Starting here, the takeaway message is, so the typical tool that one uses is, again, once again, the Whittle likelihood, right, to analyze this type of data. But of course, this may be considered limiting, in particular, if you're interested in changes over time, like the last examples that you've seen, right? So there have been methods and approaches that have tried to go beyond the Whittle likelihood. One model, one approach by Cadonna, Cottas, and Prado, which is typically based on the non-literature mixture of Gaussians, right? Although with particular choice of the weights on those mixtures, and then instead a model that tries to decompose the time series into different segments. And as you can see, we are getting really close to our times here. So in terms of, I mean, importance or interest that there is on these topics in the literature. So I hope that these may serve as a sort of introduction or interest of things that one can look at, right, in this regard. I would like to finish up, although probably, yeah, I don't have much time to go much further, but I would like to just discuss a bit a recent paper that we also did in for studying EEG data. And in particular, in this case, I wanted to point this out because the interest here is that typically, on one hand, right, the spectral analysis models treat time series from each subject separately, and they basically obtain independent spectral densities to each time series. We are considering here a data set from the Minnesota twin family study, where we have data on 557 adolescent twin pairs. And the idea here is that there are going to be commonalities probably in the spectral densities of twins, and we want to study those. So since the EEG data are collected on twins, we may assume that the time series have similar underlying characteristics. So we will want to borrow information across subjects in our estimation. And in this case, we're going to use a nested Dirichlet process model, basically, at the end, in particular, what we call, what it is, a nested Bernstein-Dirichlet prior model, to estimate the power spectrum of EEG signal. So basically, we estimate the spectra, right? And at the same time, we want to cluster the spectra across subjects, right? So this is the basic idea. So the Minnesota twin study is a population-based study of same-sex reared together male and female twins and their parents. And the idea here, we consider we have data from 365 monozygote and 192 dizygote twins from the Minnesota Twins study. And we want to explore the nature of the twin relationship to determine the amount of shared information within and between twin pairs. So common estimation methods are very fast and simple, as I mentioned. They do not allow information sharing across multiple time series. And we would like to obtain some sharing of information. For example, here you can see the EG spectra for some twin pairs. And you can see that there is the EG spectra tend to be similar within twin pairs, and in particular, more so between monozygotic twins. So the monozygotic twins. So what we want to do is to estimate the individual spectral densities and cluster similar spectra to improve inference on their features. In particular, we'd like to understand if there are neurobiological indicators that link the psychiatric disorders to genetic risk factors. So in this, it is important to notice that so if we can understand that spectral characteristics are heritable, then we will be able to link the spectral characteristics to possibly psychiatric disorders. So for that reason, we want to consider to establish heritability of spectral characteristics. By heritability in genetics, it is mentioned, it is considered basically the proportion of variance caused by genetic factors and environmental factors on a specific trait in the population. So in order to obtain and establish heritability of the EG spectra, we need to come up with a way to describe how much of the variance of the spectra that we see in the population is caused by genetic factors versus other factors. Typically, what it is done in this case, in EG data, is to summarize the power spectrum to a single measure by taking the average power within a frequency band. And typically, this is done by using the Falconer's formula, which is basically consider the correlation in the estimated individual spectral densities at any frequency omega among monozygote and dizygote twins. However, we would like to avoid basically having to do band averaging, selecting particular frequencies to compute the heritability. What we would like to do is to have a measure of heritability that consider the entire time series. And this can be done basically in our framework by considering basically the entire spectrum of analysis. And I'm not going to go much into the details because of time. Once again, I see the time shrinking fast. But as a basic idea, we are going to estimate the spectral density using a mixture of beta probability density functions. We are going to have the domain scale to line 0.5 because this is a spectral density. That's all we need to estimate the spectrum. At the end of the day, so the weights of these mixture of beta distributions are going to be determined by the realization of a Dirichlet process. And again, the idea here is to try to describe similarities across spectral density estimates. So some subjects may be characterized by similar spectra, and we want somehow to capture these similar characterization. And for that reason, we use what goes under the name of nested digital process. But again, I know that I'm going fast here, but basically the basic idea here, we have a spectra for each time series for each individual. It was just to cluster these spectra using basically a functional data analysis approach, which is driven by a nested digital process to describe the functions. Eventually, we will describe the heterogeneity of a sample EEG spectral density by partitioning the subject spectral densities into homogeneous groups. By obtain at the end of our analysis, we're going to have estimates of a spectral density, right? And instead of considering like we did before, like the Falconer's formula did before, but since we have the entire spectral density estimate, and we can also assess the variance of the spectral density estimates from our model, what we can consider is an extended measure of heritability of a power spectrum by considering basically intervals of frequencies. And this is again based on the fact that we are able to estimate the variance of each frequency across all the curves from our posterior distribution. And in particular, this interval can be also, we compute it 0, 0.5. So we can compute the heritability of the entire spectral density, that is the full spectrum heritability. So just to give an idea of the type of, so we consider just 3 channels here, CZ, O1, O2. And we have our data on male and female twins. They are all age 17, so same age. And one can see that, I mean, these are different channels that we're considering, and one can consider, can see that there are some, so each line here represents a single subject, and there are some groupings that one can see somehow in the estimate, right? So the estimate somehow follow more or less the same patterns. And in particular here, we're going to look at a few frequency bands, the delta, the theta, alpha, and beta, which are typically considered in the analysis of EEG data. And one can consider basically, and didn't go through all the results here, but one can obtain basically on one hand by looking, inspecting a bit more in details these images, right? That there is heterogeneity in the spectra across subjects, but also that some of these variation that is being seen may be driven by genetics, by actually studying the band heritability, so the interval of frequencies as we've seen before. And in particular, you can see that in some data, in some bands, the heritability is higher than others. So characteristics that are present in different subjects, right? And in particular, another important point is that one may wonder if our model was able to obtain reconstruct the information on the twin relationship. And typically, our model was able to detect the twin structure in the data, clustering monozygotic twins power spectra together for the CZ channel, for example, in 26.4% of the posterior sample, compared with 8.9% for dizygote twins and only 4.6% for non-twins. So other thing that one can consider here, but basically this is it, I think. So the takeaway message here is that there is some ongoing work in the analysis of EEG data. And here I have mentioned basically 2 possible developments. One is this idea that we are not stuck with a little likelihood, and so a more flexible and more amenable, easier also to work with likelihood may be possible to be defined. And second, the idea that one can also try and it may be interested in seeing how spectral densities change over time. So the idea of dynamic behavior, right? Also within the EEG data. And finally, also the problem from multi-subject, right? Again, comes up. Multi-subject analysis comes up again, and more complex multivariate models are needed. And in particular, the idea that there are ways to describe heterogeneity and similarities across subjects. So I think that this is, again, an area of research that has received quite some interest in the literature. And as I said, the statistical imaging community is very open to statisticians. So I would probably I'd like to invite you maybe to consider this as one of your domain of applications. I hope this was helpful. If there are any questions, I see there is a question that Mengyan has asked, but probably I will refer these to later. In particular, how does noise propagate in the Fourier space? For example, independent noise in the regional space will be correlated in Fourier space. That's a good question. I have to think a couple of more minutes about this. So probably I will refer these, maybe we can discuss later. I mean, separately. Independent noise in original space will be correlated in Fourier space. Well, you will have a particular, if I understand your question correctly, right? You will have a probably high frequency spectrum in this case, right? So I need to get back. Maybe we can discuss offline on this. And also for the second question, with the likelihood since they find a certain covariance structure from the regional space, I'm not getting it, so I'm gonna write it down and maybe we can check later on this. I'm not sure, basically, if I understand correctly. Okay, other questions? No? See, okay, yes. Okay, good. So if there are no questions left, I would like to thank you all for participating. It's been a tour de force, but I hope that you enjoyed it somehow, or I was able to provide some pointers. That was what I wanted to do. And I invite you to follow the ISBA meeting. It's going to be super interesting and super exciting. Thanks. See you soon.
Thank you.
Thank you. Yeah, we should go out. It would be nice next year in Montreal. We will be able to go out and discuss. So I'll be looking forward to meet you all in person as well. So bye.
Can I end the meeting now?
Yes, yes. Thanks.
Should end the recording.
Okay.